Hello, good morning.
Good morning. How are you?
I'm doing great. And how about you?
I'm doing well. Thank you.
Yes, I'm good.
You and the family are doing well.
Yes, please.
We thank God.
Please, how about you?
Oh, we're all managing small, small. Mm. Thank you.
Okay, thank God.
All right. So I think today we need to close a little bit early because I have some class. So even now we can only do maybe 1 hour because I have some class with some master students as well. So we'll go through this question quickly. After that, then we will, because we can't wait for everyone. I don't think everyone would like to. We'll be able to make it. I think today is a holiday. It should be fine. Okay, all right. Okay, so quick one, and then after that we will proceed. Okay, so let's see the questions that we have here. I don't know whether you finished that, but let's see the first question. Question number one. Question number one goes this way. So what is the first step of glycolysis is catalyzed by exokinase? What's the answer for that? Is it a true or false statement?
It's true.
Yeah, true.
It's true.
Okay, that's good. That's true. And then glycolysis produces 2 molecules of carbon dioxide.
It's false.
Which is false.
Yeah. If 10 glucose molecule undergo glycolysis, please mute your mic.
That's false.
False. Because 10 molecules of glucose we give what? We give
20.
20. Good.
Yes.
And in the absence of oxygen, plants will undergo lactic acid fermentation.
False. It undergoes alcohol fermentation.
That's false. And then formation of acetyl in the transition of the release of carbon dioxide.
I think it's true.
That's true statement. In a Krebs cycle, there are 4 oxidation processes.
That one too is true, sir.
Yeah, because the formation of Na, DH, and then there will be 3 of this. This all will be as a result of what? This and FAD, DH2. Okay. All right, so that's a true statement. And then Na generate more energy than this. True or false?
I think it's true.
Is it true or false statement?
That true.
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 Photorespiration reduces the efficiency of photosynthesis. Is it true or false?
That is true.
Cactus is an example of C4 plants.
False.
No, false. It's camp plant, not C4 plant.
And high concentration of ABA in the gas cell causes stomata closure.
Yes. True.
True statement. All right, good. So this is a little bit of okay, so I'm gonna start. Please come again.
I was asking, where are your ladies today?
I don't know. A lot of people are not online, but we can't wait because I have another class. So
let's see the slide for the answer. So that
I prompted them yesterday about the class.
So let's Because if I see you are going to wait to eat into the other classes, so
Eric, it will be part of the slides, okay? So don't worry.
Okay.
okay, so okay, so we have photosynthesis, and then when you come to photosynthesis, photosynthesis is more of reverse of what? Osmosis. Okay, so when we take the photosynthesis, it's more of reverse of what? Osmosis. Remember, in I say, look at me. Photosynthesis is a reverse of what? Reverse of respiration. So photosynthesis is reverse of what? Cellular respiration. Okay, it's a reverse of cellular respiration. Okay. And then so if photosynthesis, remember photosynthesis, respiration involves what? Involves what? Intake of what? Oxygen, and to generate energy. Okay, and releases what? Water and CO2 gas. And then when it comes to photosynthesis, this is okay, involves intake of water and CO2, okay, and give out what? And give out generate energy, okay, the chemical energy, food, glucose, and releases oxygen as a byproduct. Okay, so it's like so what is happening here? Okay, is basically a reverse of this. Okay. What is happening here is basically a reverse of okay, what is happening here is basically a reverse of what? A reverse of cellular respiration. Okay, whereby you take in what? You take in oxygen to generate energy and release that water and carbon dioxide. For the synthesis, you take in water and carbon dioxide In presence of what? In presence of light energy, and then you generate what? Glucose and release what? Oxygen as byproduct. So this is basically what is here. Okay, so for the synthesis, light energy goes in there to build chemical energy. All right. So up and after that, oxygen will be released as well as byproduct. And then we have this particular formula here. So we have a formula here like that. Carbon, so 6 molecules of carbon dioxide, okay, reacting with what? With 12 molecules of water, giving us our glucose, and then the what? Giving us our glucose here, and oxygen will be released as well as what? Some amounts of what? Some amounts of water. All right, good. So today, what we're going to talk about, okay, it's more of elaborate version of this. So this is the summary of photosynthesis. Now, photosynthesis is actually, when you go to detail stuff, okay, it's actually in two main processes. Okay, we have some part, we call it light dependent phase. That is where the light energy will be captured in the form of photons. And then we have what? And then the product that will be generated there will then be used, okay, will then be used to generate what? To power the dark phase, which means that, which we call the what? The light independent phase of photosynthesis. All right, so we have two main phases. We have that is the light dependent phase, which means that the part that is that can only occur when light is present. And then we have the light independent phase. Okay, the part that doesn't require light to occur. All right. So we have the light. So the light dependent phase, there are something we call photosystems. Okay, and these photosystems, they are like antennae. These photosystems are like antennae that's going to what? Capture light. They're like antennae that's going to capture light. And then at the end of the light phase, okay, which involves the capturing of light and then using the light energy to cause what? Excitation of electrons, meaning that electrons are going to move to higher energy level, that is excitation. All right, and then we have the what? We have the, and this one is occurring in the what? Now the whole of this process here, okay, the whole of this photosynthesis is actually occurring in the what? In the organelle we call what? That's the chloroplast. Okay, so chloroplast of plant cell, which we elaborate very soon. So we have the first part, that's the capturing of the light by photosystems. And then these photosystems, after the capturing of the light, okay, what is going to happen? We'll use the light to generate what? ATP and NADH, DPH. All right. So we call this one nicotinamide adenosine dinucleotide phosphate. Okay, so because there's hydrogen here, we call it what? Hydrogen nicotinamide adenosine dinucleotide phosphate. And then we have our ATP. So these are the two main products that are produced from the light phase of photosynthesis. And these two products are going to be used in the what? In the light independent phase of photosynthesis, which is also known as the what? The dark phase of photosynthesis. Okay, where there is going to be fixation of what? Carbon dioxide. So in this process, the light phase, it's the oxygen that we are talking about is going to be released at the what? During the light phase of photosynthesis. Why? Because the light will be used to, we call something photolysis of water. Okay, whereby let me go here. So we are saying that photosynthesis involves two processes. What are they? We have light dependent phase, and light independent phase. All right. And this light dependent phase, what are the products you are going to get? Here, what are the things that are involved? Here, it involves something we call photo systems. Okay, and this photo system, what do? They capture the sunlight. The sunlight in the form of photons to cause excitation. Electrons. I mean, when you say excitation of electron, meaning that moving electrons to higher energy level, because excitation of what? Electrons. And through that process, we will have what is going to be produced there. In that same process, there's something that will happen. We call it photolysis. Photolysis of water. Okay, photolysis of water is basically splitting water molecules into what? So let's say if I have a water molecule here like this, okay, will give me what? Will give me the oxygen. Okay, so that I'll get my oxygen here. Oxygen plus what? The 4 electrons released. Okay, plus hydrogen gas. So we get our hydrogen here. Hydrogen plus ions. So like that. So it is true. So the oxygen that is produced from photosynthesis is coming from where? The splitting of water molecules. And that splitting of water molecules due to the presence of sunlight is called what? Photolysis of what? Split. It's called what? So you have the splitting of water molecules into that is a CO2. I say into what? Into oxygen, hydrogen ions, and electrons. And then what are the products? The products of the light phase. The products of light, what are you going to get? You will get ATP will be produced and what? NADPH, which is known as what? Hydrogen nicotinamide adenosine dinucleotide phosphate. Okay, please don't confuse this one with this. This one is what? Hydrogen nicotinamide adenine, okay, dinucleotide. There's no phosphate here. This one has phosphate in it, diphosphate. All right. So these are the 2 products that will be produced from the what? The 2 products. Hello.
Sir, check the oxygen.
Yep, the oxygen.
Yes,
Oh, I think I've added more than two
yes.
molecules, this and that. So here should be two. So it should be this. Okay, it should be two molecules. Well done. Thank you. Good. Okay. So we have the what? So this, what is here? What I have here is a summary for what? This is a summary for the dark, the light phase of photosynthesis, which is what light dependent phase. And where is it occurring? We say that the light dependent phase is occurring where? Light dependent phase, what did I say is occurring? Yes, I said it's occurring where? Hello?
Cytoplasm.
I said it's occurring where?
Ghana.
Too much noise in the background. Light dependent phase, I say it's occurring where? Where does it occur?
In the presence of
Sir,
water.
is it the Ghana?
No, where?
Grana. Grana.
Where in the chloroplast? Yep. Where in the chloroplast?
When the chloroplast, that's the
Sir, photo system.
journal.
No, okay, all right. All right. So please, this is the summary. So I want you to get a summary right before we proceed. Okay.
Okay.
So remember, the dark phase of photosynthesis is with the photosynthesis has two phases. We have the light dependent phase, the part that depends on light. What are the things there? They have something called photosystems. What the photosystem like antenna. They capture what? They capture the sunlight. So you have various photosystems, 1 and 2. Okay. See, 1, photosystem 1 and photosystem 2. Okay. All right, like that. And what are they doing? They capture sunlight. And when they capture sunlight in the form of photons, what will they do? They will cause excitation of electrons. So electrons will be excited to move from where? That's the high energy level. Okay, so they move to higher energy level. All right, and at the same time too, the light that has been absorbed will be used to split water molecule in the form of what? So this water molecule is being splitted. So let's say here, light will come on top of it. Here, light will be here. Okay, so we need light. We need sunlight for this process to work, to occur. Okay, great. So after this photosynthetic, after this splitting of water, okay, what are you going to do? Produce what? Oxygen. So all the oxygen molecule we are talking about, okay, it's coming from the what? The light phase of it, or light dependent phase of photosynthesis. And then we also produce hydrogen ions, and then we produce what? We'll have what? Electrons. Then some of the product will have ATP and NADH to be produced. And these molecules are needed at the what? At the dark phase of photosynthesis, or the light independent phase of photosynthesis. Okay, so then let me copy that. Okay. You know, let me duplicate this one rather. Okay, save this one. Okay, so let's see the word, the dark phase. Okay, so light, so let's see the light independent phase. So light independent phase. So light independence phase, that's one. We don't need this, but we need this. And here, what will happen? We need this to build what? Okay, so this light independent phase is a cycle, and it is called Calvin cycle. And this Calvin cycle requires these two molecules here. And at the end of the cycle, what are you going to get? We'll get our glucose and some amounts of water. All right, good. So all these processes, so here does not require what? It is light independent, okay? So whether light is there or not, it will okay. Doesn't depend on light. Whether light is there or not, it will what? It will okay. So this is basically the what? The two main phases. We have the light dependent phase, which requires photosystems to capture sunlight in the form of photons, causing excitation of one electron. And as I said, excitation of electrons simply means that they are moving to a higher energy level. And then that same time, we have what? We have splitting of water molecule. The word lysis means split, but here remember is the photon. So okay, in the light phase, so photolysis of water, splitting of water molecules like that to give us this. Okay, and then in the course of the light phase, the main product here will be ATP and NADPH that will be produced, and these molecules are going to be used in the what? In the light what? Independent phase, which is also known as what? The Calvin cycle. So it was there that we use the ATP and NADH nature from light independent phase, and then to make what? Glucose. So this is the recap of the entire process, and this is the diagrammatic illustration. Okay, this is the diagrammatic illustration of the what? Of photosynthesis. So light will come here, captured by the what? Captured by photosystem 1 and 2. Okay, in the granum of the what? Of the chloroplast, and then what is it going to do? It will build up ATP and NADPH. Okay, and then after that we will have the light independent phase, which is also known as what? Carbon cycle, a Calvin cycle. And in this Calvin cycle, okay, that is where the what? The carbon dioxide will be used to generate the what? The glucose. All right, so this is the summary of the whole thing. Now let's go into a little bit of intricate detail. Okay, now so we talked about photosynthesis occurs in the world. It's okay.
Hello, Doc.
Hello, please.
Please,
I'm listening.
Yes, please, you were asking where does the light dependence phase occur?
Yes.
We couldn't understand. Please, can you help us?
Please, I can't hear you.
So please, you asked us about where the light dependence phase of the photosynthesis take place.
Yes, please.
We couldn't respond.
Oh no, we'll talk about it. It will be in the next slide.
Okay, sir.
We will answer it in the question. As I move, I think about 2 slides down, it will come.
Okay, sir. Thank you.
Yeah, so good. So we have what? We have photosynthesis, and photosynthesis is carried by plants, algae, and some what? Some bacteria. And those bacteria, we call them what? Remember, we call them cyanobacteria. Bacteria. And this cyanobacteria, they have what? They have chlorophyll to what? To absorb sunlight as well. And some other prototysts. Okay, in fact, algae are prototysts. So in fact, and then we have some special prototysts, like for instance, euglena, okay, and co. All right, great. But for the bacteria, we have the cyanobacteria group. They are capable of what? And then some prototysts like euglena. Some prototysts like what? Euglena. Okay, so some prototysts like euglena, they can all what? They have the chlorophyll pigment or absorbing sunlight for photosynthesis. Now, so plants being photosynthetic, if we are going to be photosynthetic, then you should have what? Then if we are plants and algae, you have this organelle called what? You have this organelle. And then, okay, so this organelle, that's a chloroplast. That is where the chlorophyll pigment is located. All right, but for those, for bacteria, bacteria doesn't have the chloroplast. It doesn't have the chloroplast, but it contains this pigment, contains the chlorophyll. So if you come here, the photosynthetic bacteria, which is the cyanobacteria, okay, which is the cyanobacteria, okay, the photosynthetic pigment, which is the cyanobacteria, okay, all right, which is the cyanobacteria, okay, what is going to happen is that, what is going to happen is that this cyanobacteria, okay, this cyanobacteria we have here, all right, this cyanobacteria we have here doesn't have chloroplast, but it has this pigment, it has a chlorophyll for what? For capturing sunlight for photosynthesis. Okay, so it may not have the whole organelle, but it has the pigment, the green pigment, okay, for photosynthesis. All right. So we're talking about photosynthetic pigment, and we just mentioned chlorophyll, like this one, which is the green pigment. Okay. We just mentioned the chlorophyll as a green pigment, all right, which is capable of what? Absorbing sunlight for photosynthesis. Now, it's not only green pigment alone, it's not only okay, it is not only the what? It is not only the chlorophyll that is capable of capturing light. We have other pigment like carotenoid. Okay. Okay, so carotenoid can also, so here, carotenoid usually they are able to absorb the blue and then the violet light. Okay, so they also contribute. So because they reflect, so what you reflect in like what you reflect here is what gives your color. So for instance, in the case of what? In the case of chlorophyll, the reason leaves appear green is because the leaves will what? Will absorb the red and then the blue to violet light. So if you look at the colors of the rainbow, okay, the reason why the what leaves appear green is because they reflect the green color. They don't absorb it. So if you reflect, the color you reflect is what we see. So our eyes will only see the colors you reflect. If you reflect green, okay, it means that then you are absorbing the other light. Okay. What are the colors of the rainbow? What are the colors of the rainbow? Yes, what are the colors of the rainbow? Let me take it for you here. Okay. All right. So let's see something here. Let me pick this one for you. All right. So let's see. So these are the components of the white light, like that. So we have our prism separating it. So we start from red, okay, orange, yellow, green, blue, indigo, violet. Now, if you are a leaf, what it means is that you reflect this green light and you absorb the rest. Okay, so chlorophyll like this is able to what? It's able to reflect green. So that is why leaves appear green to our eyes, because chlorophyll reflects the green light and then we absorb this word, this blue light, okay, blue to violet light, okay, for photosynthesis. When you come to the carotenoid, okay, the carotenoid says, so here, all these ones, so once you appear green, it means that you are absorbing the rest. The rest are being absorbed. That's why we can't see. Now, if you take something like carotenoid, like the carrot, if you look at this, the color of the carrot here, so it's appearing orange, okay? So this orange color here, what happens is this, okay? What happens is this. So the carotenoid gives the carrot the orange color, we've said it here, okay? Now, why is this one appearing orange? Because it's what? It's what? It's reflects the orange, okay? It reflects the orange, all right? It reflects the orange, okay? It reflects the orange here, as well as what? And then as it's reflecting the orange, it means that it is absorbing the word, the other colors. So anytime you see this carrot here, this side here is reflecting the orange color of this spectrum. So it is reflecting this. And the leaves on top here is reflecting the green. Okay, reflecting the green. Okay, so the leaves appear green because they reflect the green color of the white light from the sun. Okay, and they absorb the rest. Okay, here, so the carotenoid like this is going to, so if it appears orange, it means that it reflects the orange color. Okay, all right, so all those colorful stuff, that's the one they reflect and they absorb the rest. So if you come here, you realize that the chlorophyll will absorb the red and the blue. So here, the red part of this side here, chlorophyll will absorb all these parts, will absorb this part, all right, and then will reflect this green, okay, and likewise the carotenoid is going to do what? So this carotenoid will absorb the blue and violet strongly, and then will reflect this one. So this is the ones they reflect is the ones we see. So what do you reflect is what we are going to see. Okay, so carotenoid, like if you look at the carrots here, they also contribute, they also help to absorb what? Light. Okay, they also help to absorb light. Okay, they also help to absorb light. All right, especially so here you can see that they are absorbing the what? The blue violet components of the light. Okay, chlorophyll is absorbing the red, okay, strongly, like that. Okay, so the part you reflect is what the part we will see. Okay, good. So now let's go to the photosynthetic organelle. Now, photosynthetic organelle is the chloroplast. Chloroplast here is a photosynthetic organelle. And this photosynthetic, what does it do? So it is capable of absorbing sunlight for photosynthesis. But within this photosynthetic organelle, we have what? We have, so this organelle, what is here is the organelle. Okay, this is the chloroplast. And you can see inside the chloroplast, we have this membrane structures inside the chloroplast. This membrane structure, each of them, so each round thing here, each round thing is called granule. So each round thing here So it's called what? So we have the granule. All right. So this round, each is called what? Granule. Okay. Granule. And this granule here, the granule here, as you can see, the cluster. So this each one is granule, and they are made up of membranes. They are membranes, okay? The membranes here, okay. So we have this all right. So you have all these membranes there, like so this cluster. So this cluster here, all this cluster is the granule. Okay, so this cluster, all the cluster like that is granule. And each also this round thing, round here like that is granule. And so 1, 2, 3, 4, 5, 6, 7, 8, 9. These are grana, okay, which is the plural. Okay, grana. That's the plural of the word granule. So each this round, all this 1, 2, 3, all this stack round together is called what? It's called granule. Okay, so these are the granule, 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 granule. Okay, so all these ones are granule. Okay, and then collectively we call it what? Grana. But each round, the small one, let me use the marker. Let me go for drawing. Let me come and draw. So let's say now each one of these. So each one of these here, oh, look at me. Each one of these, just one these. Okay, that's the pack, this stack here, this. So this one, each one of this small round thing, this, the whole of this is the granule. Okay, the whole of this granule, and all of them, the plural is what? Grana. And each one of these is called thylakoid disc. Okay, so all these ones are made up of membranes. So which means that from here we can see clearly that the what? The gran, what is forming the gran in the collection of the thylakoid. Okay, this is equal to the what? Is equal to the granule. And then several gran, okay, a number, several granule gives us grana. So the whole of each one is granule, and 1, 2, so several of them is grana. Are we there? Okay, great. So this is, and this liquid. So here inside here, we have this liquid component. So the liquid component here, this liquid component here, the liquid component we have here is a what? It's called stroma. All right. So this is my granule. This round is my what? My granule. Okay. And then the what? The liquid component here is a what? Is a stroma. Okay. And this granule consists of what? This of what? A stack this of thylakoid. Thylakoid. Okay. So each one Each one here is a thylakoid. So this structure is the chloroplast. So inside here are these are the grana, all of them grana, and then each one here is granule. The granule consists of what? Stack of thylakoid disc. And then this liquid component is called stroma. Now, when it comes to the first part, what I talked about, so we have who is going to capture the light. Now the chlorophyll, you can see this is looking green, 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 green. Okay, the thylakoid are looking green. Okay, so those thylakoid, that's where the chlorophyll, okay, the pigment chlorophyll, this pigment chlorophyll is located. Okay, that is where the chlorophyll is located to capture the sunlight for photosynthesis. So this side is where the chlorophyll is actually located. So the light phase of photosynthesis, okay, the light phase of photosynthesis, the light dependent phase of photosynthesis occurs within the grana, okay, that's within the thylakoid, this one. Okay, while the light independent phase occurs in the stroma. Okay, so the light dependent phase is occurring here, and light independent phase occurring here. Okay. Any questions so far? Any questions so far? Any questions so far? Okay, so we know that inside here, inside this thylakoid, the thylakoid is 1111 like that, as I showed it here. So this one is thylakoid. So the class of the thylakoid gives us a granule, and all the granule here together gives us a grana. Okay, the plural of granule is grana. Okay, so within the thylakoid membrane, that is where the pigment is located. Okay, within the thylakoid membrane, that's where the pigment is located. All right, and the pigment location of the pigment, that's a chlorophyll pigment for absorbing sunlight for what? For photosynthesis. Okay, that is where the pigment is located for absorbing sunlight for photosynthesis. And then we have the what? We have the stroma, which is the what? The liquid component. And the liquid component, that is where the what? The light independent phase of photosynthesis occurs. So light dependent, that's a granule, and light independent, okay, occurs in the what? Occurs in the stroma. And remember, what is forming the granule is a what? Is a thylakoid. Okay. All right. Great. So yeah, back to this one. So yeah, so we have so like what I said earlier on, the light independent, that's where it's inside this thylakoid here. It has a granule here, the thylakoid membrane. That is where the light the light dependent phase is going to occur. Okay, and then the light independent phase will occur in the liquid component, which is the what? The stroma. Okay, this one and this one. So within the thylakoid membrane, okay, that is where the light dependent phase will occur, and the light independent phase will occur in the liquid component, which is the stroma. Okay, so this what we have here is the micrography of what? Of chloroplast. Then let's talk about a little bit about what is going to happen in photosynthesis. So basically, what I said earlier on, during photosynthesis, what happens? Remember, we have water as our raw material. We have the sunlight. Okay, the sunlight will hit on this word, on these within the thylakoid membrane, will hit on this. Okay, and when it hits on this, we have something called photosystems. And this photosystem, what are they going to do? Will capture the sunlight, and when it captures the sunlight, okay, it will cause what? This sunlight will cause excitation of what? Electrons. And this excitation of electrons, okay, we move from one state to another. And in the course of that, we will produce what? ATP and NAD pH. All right. And then where is the hydrogen ions coming from? Please, where is the hydrogen ions coming from? Yes, where is the hydrogen ions coming from?
The water.
No, what process? By which process?
It comes from the splitting of the water molecule.
Yes. Okay, so that's what's the name for that process? What is the breaking of water? Yes, you are right. But what is the specific term for it?
That's
Photolysis.
photolysis of water.
Excellent. So that's the photolysis of water. So this our hydrogen ion here is coming from the what? From the photolysis of water. Okay. So here,
Of water.
excellent. So here, the capturing of the light by the photosystems. Okay, and then this capturing of the light by photosystems will cause what? That is a excitation of electron, and this excitation of electrons will be used to build up what? NADPH and ATP. And then you see that when it is built up here, they are going to be released into the stroma, the liquid component, where the dark phase or the light independent phase of photosynthesis is going to occur. All right. And then in the process, remember, during the photolysis of water, oxygen will be released, and the oxygen is coming from the what? The water. Okay, the breaking of water. Okay, will release hydrogen gas, hydrogen ion, sorry, and then we'll release oxygen gas. Okay, good, and then electrons. Okay, good. And then, so when it gets here, so inside, when this one happens inside the thylakoid here, thylakoid of the granule, this will come here, and this one is going to be used, okay, in presence of what? Carbon dioxide, and then to build 3 carbon molecule here. Okay, that's 3 because it's 3. That's what we call that the tribes phosphate. Okay, 3 carbon molecule. All right, and this 3 carbon molecule will go on to be used to build what? Glucose and other organic molecules for the plant. Okay, this is still some of the summary. Now, so quick one here. Let's take something like this. So we know that during the light phase of photosynthesis, what is going to happen? What is going to happen is this. So we have the 3 carbon molecule here. Okay, the 3 carbon molecule. All right, that is going to be what? Generated. And this 3 carbon molecule, there's various fate of them. They can be used to build glucose, which is the primary product. After the glucose can be used to build cellulose. The glucose can also be stored in the form of starch. The glucose can also be used to convert into what? Fructose. Can be converted into what? Can combine with other sugar, or can converted into other sugar such as what? Fructose here. All right. This is the sugar in what? Sugar in that is fruit. Okay. So when you take like fruit, if you can see the name here, so this is the fruit. You can get this one from fruitose. All right. So that 3 carbon molecule that will be generated. Well, the primary one is glucose. And when glucose is generated, glucose can be used to form what? Cellulose, can be stored in the form of starch, can be converted into what? Fructose in the case of fruit. Okay, fructose and sucrose in the case of fruit. Remember, you get the sucrose here in the sugar cane. Then sometimes can also be used to undergo other complex processes to build up what? Some lipids and amino acids. So which means that this primary product is very, 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 very important. The first, this 3 carbon product, okay, could be used to build glucose, which is the first step. When the glucose is built, the glucose can be converted, okay, can be stored or can be used to build components of cell, like what? Like cellulose for the cell walls, can be stored in the form of starch, can be converted into what? Into fructose, which is found in fruits, and can also be used to build other complex molecules, okay, like lipids and some amino acids. All right, great. So now that we've given the general overview, now let's go into a little bit detail. All right. Now, so we started by saying that okay, we have something called photosystems. Okay, please let me pick a call quickly for this my master student quickly, and then I'll get back to you. Please, just a minute. Let me pick this call quickly. Hello. Good morning. Yes. All right. Okay, so I apologize more. Okay, so then I need to pick that call quickly. Okay, so we are going to a little bit detail of what happens. So we started by saying that, so these photosystems, where are they located? These photosystems are located in the what? In the membranes of the thylakoid. Okay, so these photosystems, so I said the photosystem, they act like what? They act as like antenna. Okay, and then these photosystems, they are more of complex proteins, okay, located in the what? In the thylakoid. The thylakoid of the what? Of the granum to be specific, okay, all right, of the chloroplast. Okay, what are they trying to do? They will capture the light like antenna. Okay, they capture the light like antenna, and then they use it to convert what? They convert the light energy into what? Into ATP and ANDPH. Please, it's not ANDH. AND, this one is what? Hydrogen nicotinamide adenosine dinucleotide phosphate. There's phosphate here. All right, good. So here they will capture the light and use it to produce what? ATP and NADPH. And this is occurring in the what? In the thylakoid, the membrane of the what? The thylakoid membrane, okay, of the granum, okay, of the chloroplast. All right. And then you remember, as I said, this is associated with what? These photosystems, they are the main components of the what? Of the light dependent what? Phase. of the light dependent phase of photosynthesis. Okay, all right, light dependent phase of what? Photosynthesis. Okay, so what do? They absorb the light, the photosystem will absorb the light, and then they'll use it to cause what? Excitation. So this is what I mean by excitation of electrons. Excitation of electrons. Okay, that's a raising electron to higher energy level. Raising electrons to higher energy level. Okay, so that is the word. That is the excitation. Okay, raising electrons to higher energy level. Okay, that is the excitation. All right, that's the excitation. Now let's see what exactly that happens. Now these electrons, these are photosystems. There are two of them. So here I'm going to be very specific. This side we call it photosystems 2, and we call this side photosystem 1. Now let me explain this thing to you. Okay, let me even go and get this thing for you to see something here. Let me quickly get it online and just get something for you right now. Okay, let's see. Okay, let's see. Good. Let me get you something. Okay. Now, okay, so oh, all right. So look at what I have here. Now, so this is more or less light. Okay, we have something called electromagnetic spectrum. And the electromagnetic spectrum is this long thing at the top here. And as part of the electromagnetic spectrum, there's this small section we call it visible light. And this is the visible light that enables us to what? See. So this is the visible light here. Okay. So this is the electromagnetic spectrum from gamma ray all the way to the radio wave. Okay. And this electromagnetic spectrum, we have the word. We have it is moving from here all the way to this side. So the part of the electromagnetic spectrum that our eyes can see, this is the visible light spectrum. And that visible light spectrum, you see, is between what? It's between 400 nanometer to 700 nanometers. Okay, so you can see here 400 nanometers to what? To 700 nanometers. So these are the lights we can see. Right from the what? Right from the violet to indigo all the way to red. All right, good. So you can see that what we have here, these are wavelength, the wavelength of the word of the light, different colors of light. So here, the red light that it is in word is in 700 up to the end of it. So all this stretch, okay, so you can see it fades. There are various fades all the way to the end. Okay, so to the 700 nanometers, this is the wavelength of the word of the light. Okay, you can see that when you get to this side, 650, 600, 5, 500, all the way 450 up to 400 here. Good. So these are the wavelength, okay, spectrum of the word of the visible light. Okay, wavelength spectrum of the visible light. And let's see what is going to happen. Okay, of the visible light. Let's see what is going to happen here. Now, if I come here like that, so here, if you have this first one, okay, the photo systems, okay, all right. Now, what this photo system is going to do is that we have photo system 2 and photo system 1. Please, this one, the photo system 2 and 1, here, the 2 here and then the 1 here, all it means that they discovered the 1 here first, okay, and then they discovered the 2 here second. All right, but in when we come to the photosynthetic process, it is even the photosystem word, the photosystem 2, that's the light capturing starts. Please, are you with me? The 1 and 2 here is just because if here is called photosystem 1 because this one was discovered first. Okay, the photosystem 1 was discovered first before the photosystem 2. Okay, so this is the photosystem 2. Now you can see that the two of them, you see, look at that. All of them here are located. They are all located in the what? In the thylakoid membrane. Okay, all these ones are located in the thylakoid membrane. But look at something. See, this one absorbs light at the what? 680 wavelength. Okay, this one absorbs at what? At 680 wavelength here. Okay, absorbs at a 680 wavelength. All right, 680 wavelength. Okay, good. All right, so please, a quick one. Please, when you get them, just send me WhatsApp message, okay? Because I'm in the midst of it online. That is fine. That is fine. Okay. All right. So as I was saying, please, sorry for that. So here, the photosystem 1, okay, the photosystem 1 is here, was discovered first. Photosystem 2 was discovered second. But anytime when it comes to the light capturing process, the first thing that is going to happen is the photosystem 2. Rather, okay, that is where the action starts. Okay, and let's see what happens here. In the photosystem 2, the light, it will capture the light, and this light is going to do what? It's going to cause the excitation, so it will hit on this one. Remember, look at part it's going to absorb. It's going to absorb the light, okay, the light in the wavelength of what? So you can see that it is absorbing the light somewhere here in this region. Okay, if you look here, look at that. It's absorbing the light in what? 680 nanometers. Okay, 680 nanometers. All right. So 680 nanometers. That is where the light is going to be. What? That's where the light is going to. That is the section of the light is going to absorb. So photosystem 1, 680 here. So this is somewhere here. Okay, this side. That's where it will absorb. Okay. And when it absorbs the light, what happens? When it absorbs the light, what happens? It is used to cause the excitation. So it will hit here and use it to cause the what? The excitation of what? Of electron. And when this electron is excited, there will be some protein complexes within here, between here like that. And these protein complexes will be, so they will pass it like the way as it happens in the case of what? In the case of cellular respiration, the electron transport chain, similar thing will occur. So when the electron, when it hits here, the electron will be what? Will be excited. And when the electron is excited, it moves. And then as the electron, it rises from this the surface here, and then it will be what? It will move to the protein, all right, to move up. And then as it moves, it will be passed on from what? From one protein complex to another protein complex to another. It will go all the way down, and then, okay, look at what is going to happen. Now, have you seen that? Okay, so let me come here. Let me show you something here. All right. Now, your electron has been taken away from the place. Okay, electrons, okay, let's say E. So electrons here, okay, electrons has been taken away like that from the photosystems. Now, you see, in nature, if you can't just take electrons like that. When you take electron from something, It must be replaced. Okay, so you are here. You've kicked out the electron from this surface. The electron has risen up like that, and then the electron will be passed on from one protein complex to another. So one protein complex here, another protein complex, and then as it is being passed on from one protein complex, the energy in the electron is what is used to to generate the ATP. Okay, so this is more of the summary of it. So the energy in it is what you use to generate the ATP here. Okay, all right. As the electron is being passed on from so it will rise up and it will be passed on from one protein complex to another like that. Okay, and then the energy in it is used to generate ATP. Now, look at something here. On this side here, when this electron leaves, it must be replaced. How are we going to replace the electron that has left this place? Yes. Where do you think, where are we going to get the electron here to replace this one? The one that has left. What should happen? Yes. Any idea from the audience? You've taken electron away. All right. How are we going to replace it? Okay. How?
Is by photolysis.
Good. Excellent.
By
So here,
photolysis, yeah.
exactly. So now we see where the photolysis is
Water
important.
must be splitted.
Good. Exactly. Well done. So what is going to happen is that when the electron here, the photo system, okay. So when we have the photo system, okay, so the excitation of the electron, okay, the excitation of the electron will create what? Will create, so that when electrons they leave, okay, excitation of what? Of the electron, like that. So when this electron leaves, they will create deficits. Okay, you understand that? So there will be electron deficit, and this electron deficit will be paid by who? Photolysis of what? Photolysis of what? Of water. Okay, photolysis of water will split two molecules of what? Two molecules of water, like that, okay, and give what? Oxygen out, plus what? Hydrogen ions, plus 4 electrons, like this. So this electron here, these electrons that will be produced from photolysis of water will come and replace this one, what's left here up. Are you with me? Any confusion so far? Okay, so in nature, anytime there is deficit, the deficit must be paid. And how is it going to be paid? Here we have the electron. Okay, the sunlight in the form of the photons, okay, the photosystems in the in the thylakoid, okay, of the granule, okay, has absorbed sunlight in the form of photons, hitting the surface of it. Electron excited, they will rise to energy level, and as they are descending from being passed from one energy level to other, they release energy in the form of ATP. So the high energy in the electron will be used to generate ATP here by the photosystem too. But this deficit that they have created on this surface of the photosystem should be paid. And who is going to pay that electron? That. Then they will use that, okay, so that the light energy again will split. So here, light energy, let me put light energy here, the light energy, okay, the solar energy, okay, the light energy, okay, all right, will be used to cause what? Photolysis of water, to produce oxygen, hydrogen ions, and that. And to this one, that will come and pay the debt here. Okay, good. Now, at the same time, the surface of this one, of the photosystem too, will also be what? The photosystem 1 will also absorb light at this very end. You can see, look at that. It's absorbed light here at the very end here, okay? All right, it's absorbed the other light as well, okay? It's absorbed the blue and stuff like that, but where we're talking about the major concentration of the light. So to absorb the light, the high at this end, okay, 700 here, nanometer, all right? And then will cause that light, see the sunlight is also hitting there as well. So this one will cause what? Will cause this electron here to be excited. Okay, the electrons on that surface to be excited like that. So when they're excited, they will also what? They will just move up like that. And then when they move up and they are descending, this photosystem 1, as they excite the electron, and then they will use their energy to build a what? NADPH. But look at something here. The electron here, who is coming to replace it? The electron that excited here, who is coming to replace it? Look at the diagram carefully. In the case of the photosystem 2, we have to split water to come and replace this one. But in the case of photosystem 1, when the electrons are excited by the sunlight, the photons, the light photons, and then when they are descending, they'll use it to build this molecule here. Now, who is going to replace the electron here? Yes, the electron is coming from where? Yes. Look at the diagram carefully. The electron is coming from where? The electron that are good. So the electron that move from here up, after it is being passed on from the protein complex, like down, then it will come and replace the electron in what? In photosystem 1. Okay, so the one in photosystem 2 that will start the process will be replaced by what? The electron from the photolysis of water. And then the one in photosystem 1 will be replaced by the electron from the what? That electron, that excited electron here, as it descended, it will be captured to come and replace the what? The one in what? Photosystem 1. Okay, so this is basically the process that came to what? That came to occur. Okay, this is basically the process that came to occur. We have the what? We have the photosystem 2 will absorb the light, cause the excitation of electron. So as the electron get high energy level, they arise, and as they pass on from what? One protein to other. As they do, the energy in them is used to generate what? ATP. And then the electron will be captured by the what? By the photosystem 1. Okay, because photosystem 1 will also be excited. But when photosystem 1 is excited, the capturing of its electron is not done by the replacement of its electron. It's not done by the photolysis of water, but it's done by the what? The excited electron from the what? From the photosystem 2. That will come and replace the electron here. While that of photosystem 2 will be replaced by the photolysis of what? Water. Okay, and then so to go up like that, and then get here. Okay, go up like that, and then get everything here beautifully. So that is that. So that's a summary of everything here. Now let's see the pictorial illustration of what is going on here. Okay, now pictorial illustration of what is going on here. So let's say we have our photosystem 1, photosystem 2 here. Okay, so this is our photosystem 2. Now this photosystem 2, what is it going to happen? It will capture the sunlight, and then the electron here released. When we release the electron, what is going to happen? This electron will now be passed on from where? Will now be passed on into the where? These are the proteins within the cell membrane. Okay, these are the protein complexes within the membrane. Okay, I said the cell membrane, within the membranes of the thylakoid. So they will pass this electron here, and then as they pass on the electrons here, the energy in them will be used to what? Will be used to pump what? Hydrogen ions. Okay, so you can see that. Look at the energy in them. Remember here. So you see, this is this. Okay, this is the electron excited. It will be passed on into the what? These protein complexes, various protein complexes. And during that time, at the same time, photolysis of water will occur. The electron from the photolysis of water will go and replace the excited electrons here. Okay, the electrons that were excited here, they will go there to replace it here. All right. Okay, this is the excited electron. We'll go there and replace the excited electrons here. You can see that. You can see from the photolysis of water has happened here, splitting water molecule, giving you oxygen, hydrogen ions, and then the what? The electron here will go and replace the what? The electron deficit in the photosystem 2. And then this electron from the excited electron were passed on like that, okay, through this protein complex, okay, like that. So as it's being passed on like that, the energy in it will be used to pump these hydrogen ions through ATP synthase, okay, from the lumen of what? From this thallicoid space, all right, okay, through ATP synthase to generate what? Energy in the form of what? ATP. So as the electron is being passed from here, like that, that's being passed on from one protein complex to other. This, you can see some three let me project it well so that you see it well. Okay. Okay, like that. Okay, so you can see here. Okay, you can see here. All right, on this side. Okay, water splitting. This is the photolysis has gone up there. Replace what? Replace the electron that disappeared from the top here. Replace the electron that disappeared from the top there. Okay, like that. Simple. Replace the electron that disappeared from the top there. Beautifully like that. And the electron that actually excited as it descended, the energy in it will be used to what? Will be used to pump. Okay, will be used to what? Force this hydrogen ions here through the what? Energy synthase. So the energy here, have you seen? Look at that. Have you seen this arrow here? So this blue arrow down here, okay, is the energy in it that will be used to what? To pump the hydrogen ions through the ATP synthase, like a turbine. You know ATP synthase, remember? Okay, when we're doing this thing, the electron transport system, we used to pump the ATP synthase. All right, and when you pump the ATP synthase, okay, when we used to pump the ATP synthase, okay, we used to what? We used to generate the what? The we used to convert ADP, adenosine diphosphate, okay, to what? To the ATP. All right, and then you can see that. So this is the first part. That's what is happening in photosystem 1, and then this electron here at that same time in photosystem 2. That same thing, photosystem 1, the excitation will also pass the electrons through a complex. But in this case, what is happening here is that it will lead to the generation of what? Of NADPH. All right. Okay, so to just go like that, and then there's an enzyme NHDPH reductase. So here, reductase means that, remember, when you are reducing something, means that you are adding hydrogen to it. Okay, so you can see that in the case of the second part of the reaction, they are doing what? You see, they are adding this NADP plus, they are adding hydrogen to it to get what? NADPH. All right, so okay, before that, okay, so you see, I've deliberately left those terms out. I will come to it very soon. Okay, I'll come to it very soon here. So you can see two processes are occurring here. In photosystem 1, Okay, in photosystem 2, okay, it is leading to the generation of ATP. The electrons will be passed on, and energy within the electron, the excitation of the electron, is used to do what? It's used to pump hydrogen ions through ATP synthase to generate ATP. And in photosystem 1, the energy that is released from the excited electrons is used to reduce NADPH. They will use it to add hydrogen what? Hydrogen ions to what? To NADP plus to get NADPH. And remember, anytime you add hydrogen to a molecule, we say that you have what? You have reduced the molecule. Please do remember that's reduction. Now, you can see that in the case of the first one, we see you have something you call non-cyclic phosphorylation. Remember, phosphorylation means addition of what? Addition of phosphate. And addition of phosphate usually results in what? Generation of what? Energy. If you remember, anytime there is addition of phosphate, there is what? Generation of energy. Now, you can see here in the first round, okay, in the first round, there is no word. It is not cyclic. It doesn't go round, okay? There is no cycle here. Look at what is happening. The photolysis of water is here. Electron is there. Look at that. If you look here, energy hits here. Electron excited. The same energy will hit on the water here. It will come and replace it. And then this electron is passed on here, passed on onto the complex. The energy in it is used to pump the hydrogen ions here to generate ATP. And this electron goes to the what? To the photosystem 1 to replace it, to replace the what? The lost electron. Okay, and then the what? The energy, this excited electron here is used to what? It's used to convert what? Na, the energy there is used to build what? It's used to reduce Na what? NADP plus, okay, into what? NADPH. All right, are you there? NADPH. And this NADPH, all right, you see that we've also in this case, look at something here. The cyclic electron lost from photosystem are replaced by the one in photosystem what? 1. Have you seen it? Okay, so you see here, the cycle doesn't go round. In the case of the first one, it's non-cyclic because what is lost in the case of what? So the first phase, the one that's happening in photosystem 2, is non-cyclic because what is lost in what? In photosystem 1, in photosystem 2, will be replaced by that of water molecule. But in the case of the cyclic phosphorylation, the electron loss in the what? In the electron loss from the photosystem 1 is replaced by the what? It's replaced by the what? The electron that has lost from photosystem 2. So here you can see that, okay, the electron from photosystem 2 will come and what? Replace this one here. This one. You can see from here, the electron from photosystem 2 will come and replace it here. Okay, all right, okay, and the ATP is generated, okay, all right, but not what? NADH. Are you there? But not what? NADH. All right, okay, good. Let's see something here. So let's see this picture here. All right, let's see picture here again. You see my photosystem 1 goes like that. Energy in there comes through this one here. The electrons here goes here. All right, this energy here is pumping this one down. This one here goes there. This electron goes here. This complex here, the this electron here is used to reduce what? Reduction. Remember reduction. So reductase. So the enzyme, this is the reductase. The energy here is used to what? Convert the NADH2 plus, okay, NAD plus into NADH. All right, great, good. So this is the chlorophyll, higher energy level of the electron. Okay, all right. Okay, look at that. Then we have the transfer of what, electron transport chain, whereby the electron is transferred here to the thylakoid. Okay, so the electron will be transferred. Okay, from here, as it's excited, the electron transport. Okay, as they transfer, the energy in them will be what? Will be lost, and that energy is used to pump the hydrogen ions here. Okay, all right, good. And then, okay, all right, and then the ADH plus here carries out the word. So here, look at that. So when we saw the two products, look at the products here. You look at the two products. So you see the products here, we have the ATP here produced, and then we have the NAD. You see, anytime they are produced, look at all this membrane here. This is the thylakoid membrane. Okay, and outside there, this outside there, that is the word. That is the stroma, the liquid component. Okay, so you see that here. Remember that. So this NADP plus is what? It's going to carry the word, the hydrogen. Okay, so to this one here, so this one here combined with these hydrogen ions here to form NADPH. All right, and then while the ATP formation is by what? ATP synthase, the NAD plus P plus what? Reductase is the one that is going to ensure the conversion of what? NAD plus, okay, into NAD plus NADPH. Okay, and remember, as I told you in the last 2 weeks, okay, addition of hydrogen, okay, is called what? Reduction. Okay, so here you hit there, electron hits here, excited, the same heat energy, photolysis of water, electron will move here, go and replace this one. This electron from here is passed on onto the series of what? Protein complexes. Okay, that's the electron transport chain. Okay, they pass on from this, that. Okay, the energy in you is there, is used to pump the what? The hydrogen ion through the what? ATP synthase. Okay, to generate what? ATP. All right, and then, all right, we have the what? We have the light, the photosystem one. Pop. Okay, so it goes on like that. So the electron here will also be excited. When it's excited, okay, to pass to the complex, this complex here, and then this one will convert this particular complex here, which is the NAD plus, will convert this reduction process by reducing the what? NADP plus into NADPH, which is the what? The hydrogen carrier. All right, good. But its electron, there's the photosystem one, its electron will be replaced by what? By electron from the what? From the photosystem two. Okay, good. Are you there? Any headache so far? Any headache so far? Any headache so far?
No, please, Doc. No, please.
Okay, great, excellent.
Doc,
Good. Yes,
Doc,
please.
please highlight on the electron replacement for me.
Okay, good. So let's see something here. Now, let's take home. Let's take home that photosystem one and photosystem two. Okay, so let's say you are the man of the house. okay, and your wife, so you are the photosystem 2, and then your wife is photosystem 1. Excellent. So let's see what is going to happen. Now you work. Let's assume that your wife is a stay-home mother. Please, are you listening to me? Hello, are you with
Yes,
me?
please, Doc.
So your
Yes,
wife,
please. Yeah.
Yes, please, Doc.
Yes, it's a housewife. It's a housewife.
good housewife. Excellent. Okay, so it's a housewife, and then you are the word. So housewife, you are here. The housewife is photosystem 1. The two of them, the 1 and 2, is just because your wife was discovered first. That's why your wife is called photosystem 1, and you are photosystem 2. Okay, what will happen? Okay, now what will happen is this. So you get energy. Okay, you get energy. So your wife is a housewife. Now your wife also works small. Okay, so even though it's a stay-home mother, a housewife, maybe that's some little business at home. Okay, so little business at home means that sun also shines upon her. Okay, so let's see something here. So you are the man of the house here. All right, so the man of the house, what is going to happen is that the sun hits on you, you work. Okay, your wife is also working, small stay-home mother, will have maybe some small business at home, so he takes care of the children and then manages some small business at home. Okay, so when the sun hits on you, the business that you go to work, you get the money. Okay, now how do you get the money? So the money you get is in the form of what? So the money you get that hit on you, so the money you are getting is the heat energy from the sun. Okay, so the heat energy from the sun will hit on you, and when it hits on you, what is going to happen? It will cause excitation of what? Of electrons. So you get the money will be generated from you. So the money, that money is being will excite you up. That is the electron here up. Okay, good. And then when this money, you get this money, which is the more or less the energy that you've also acquired in the form of what? Electron, which is the money, the currency you have now. Then of course you are going to use it. You also need to survive. So you use some of the money to buy food, use some of the money to buy clothes and stuff like that. So use some of the money to buy food, use some of the money to buy clothes, and then use some of the money for school fees and stuff like that. Now, okay, buy clothes, pay rent and stuff like that. So as the money, the moment you receive the money, as the money is coming down like that, so the money is coming down. Okay, as the money is coming down, passing through those protein complexes, school fees, rent, clothing, the food at home, all these ones, okay, like that. Good. Then how would that money will be replaced? Then you also have what? As you do that, as that electron here has been excited, okay, then we have the government here. This is the government. The government here is the water. But why is the government? Now the government also makes money. Okay, how is the government making money? Okay, so the government is going to mine gold, manganese, the sea, export what? Fishes from the sea, the mines, everything. So those money, the government's money here, this is the government here, that same stuff, that same heat stuff, resources will be used to split the what? The water here, okay, which is the minerals, our water resources and stuff like that, the gold, our bauxite. And then, so this is the government here. So government will generate the what? The money in the form of what? In the form of electrons here. So anytime when you leave, the ones here lives here and create deficits here, the government will pay you. Okay, we'll come and replace this money for you here. All right. So the government here, the government in this case here, okay, from this my analogy here is the water. So heat will split on the water too, and then the water from here will come and replace your money here. But your housewife, when the business money is short, okay, because the sun will also hit on him, such as life, sun will hit on her as well, and then this money, when the money becomes short, your money from here, the money paying school fees, the remaining will be captured by her here. Okay, so her money, so with the money that she is using for business, when it goes like that, those one will be replaced from the one coming from you. Okay, so you are the man here, you come and replace hers. Okay, so your money here is used to build energy, ATP here. Hers is used to cater for home. So let's say the home stuff here is the NADPH, and then your own is used to build the ATP. Okay, but when hers is short, that short of electron will be replaced by you. Please, you get the analogy here. So you are taking it from the government here. So it's a life. The sun is hitting both of you, all right. Okay, everybody is suffering. Whether you are a man or a woman, you are suffering. That's true. Okay, your suffering here is being what? It's being cushioned small by the government by paying you. Payday will come. So when the payday comes, the electron here excited, okay, goes. So when you excite your electron, when your money is finished from here, that deficit will be replaced by photolysis of water. That will come and replace your electrons. But in the case of your wife, when the life reality lashes her, okay, when life reality lashes her and her electrons are gone, you will replace that. The electron that came from you will replace hers. Are you there?
Yes, please, Doc.
Excellent. Good.
Thank you, Doc.
So good. So here you can see that the wife is building this molecule here. Okay, this building this molecule here. All right, building food and taking care of the kids and stuff like that. So the wife is generating this on this side. Okay, but her electron is replaced by you. While in your case, the energy you actually generate from here is used to build what? It's used to build ATP. From what? From chemosmosis. I think you are familiar with this word. It's used to build ATP here. That's energy here. So in the house, everybody is contributing to sustain the house. Okay? Because you need to build future together. So then while the man is building ATP, the wife is building NADPH. But anytime the housewife is short of electron, you replace the electron for her. Okay? All right. So this is simple analogy. So we are getting ATP from here, while we are getting NADP from the wife. So photosystem 1 giving us NADPH, while photosystem 2, which is giving us what? ATP. And this ATP and NADPH are what is going to be used in the what? In the light independent process. Okay, so this is how two of you are going to build the future. So what is the future we are talking about in this context? What is the future I'm talking about in this context? Yes, the future we are going to build. What in this context? What am I referring to as future in this context? Yeah, the future stands for what in this context?
The light independent phase.
Excellent. But what product will be built in that future?
Glucose.
Glucose. Eh.
Excellent. Do you get a point, right?
Mm,
So we
yeah,
start the life
yeah,
cycle
yeah.
here. Then in the future, now our wife is contributing this,
But
husband
glucose.
contributing that, and then in the future, okay, in the future there, where we don't have, in the future, the unknown future, which is the what, the Calvin cycle. Okay, all right, the Calvin cycle here. So the future is in the stroma. Okay, here in that future, we need energy. We need this hydrogen carrier. Okay, we need them to build the future together. And that future is going to be the glucose, the final product. That's the glucose we are going to build there in the future. All right, good. Okay, so here we have our synthase here generating our ATP, while this one is generating our NADP. So here the wife is not generating ATP, but it's generating what? NADP, which is what? Which is a hydrogen carrier. Okay, like that. It's transporting hydrogen. Okay, all right. So NADPH is being transported. The wife is generating this particular molecule, while the man is generating this. Okay, good. Are you there? Okay, all right, good. So now, after we've done this at home, now what do we need? Now we are now going into the future. So this is the struggling phase. The struggling phase when the light is equipping all of us. When the light was hitting us. When the light was hitting us. When during the struggling phase of our life. Okay. All right. During the struggling phase of our life. Okay. When the light is tripping all of us together. Okay. That is the struggle phase, the life dependent phase. Okay, the pain of the school fees, the crippling of the trading of the wife at home and taking care of the kids, paying the rent and stuff, something like that. We are all doing this so that we can build a house. We can build a future. Okay, we can build something. We can build wealth. We are going to build wealth together. Okay, so now that the life has really hit us hard, now let's see how the future is going to be like. Okay, the future where we are out of the world, the light. Now the light is not hitting us anymore. Okay, so when the light was hitting us, what happened? Okay, the government paid the husband. Okay, replace that money all the time. Okay, when the wife's business money short, the husband replaced it. So the wife was able to build the kids here. The money, fuel is coming from the ATP. So NADP. So we need this. We need the ATP here, and we need a word NADPH here. We need it badly. We need both badly. And whilst we need this one, okay, what is going to happen? To go and build a future that does not depend on what? The light. Are you there? So we are going to that future. Now this is in the future now. The light independent. Now the light is not going to hit us. Okay, like that. Why? Because we build the things we are going to use here. And remember the things we built. Okay, so here in the light dependent phase, let's go back here. Now we're staying in this place. In the light dependent phase, we're staying here. We're staying in this rented apartment here called what? Talakoid. And that is where you were able to pay, you were able to generate the savings, the ATP, and then these are the things we're buying. We're able to buy some raw materials here, paying the school fees and stuff like that. So we generated all these two things. Now we head into the future where our life is, where the sun is not going to hit us. So in the future, what is going to happen? In the future, there is this beautiful cycle. This beautiful cycle is called what? This beautiful cycle is called Calvin cycle. And what is this Calvin cycle going to do? what the carbon cycle is going to do is that this carbon cycle is going to absorb carbon dioxide. Okay, carbon dioxide is going to use the carbon dioxide, and then there is this 5 carbon molecule. Are you with me? There is this 5 carbon molecule called that is the ribulose bisphosphate. Okay, this beautiful 5 carbon molecule, and as a matter of fact, this 5 carbon molecule here is more or less the most abundant. It's more of the most abundant molecule. Okay, one of the most abundant molecule on the planet, because almost every plant has this particular molecule. Okay, please observe this one. Somebody is knocking at the office. So let me quickly just pick one. David. Abby. Yes. So we have the what? We have the light independent phase. In this light independent phase, we have the ribulose bisphosphate here. Okay, and this just like the way in Krebs cycle, we're having the oxaloacetate here. Here we have 5 carbon molecule here, and this 5 carbon molecule will be what? Carbon dioxide will be added to it. Okay.
Hey,
Hello.
I can't see the slide you're talking about. Or is that my end here?
Maybe it's your end. Please, can you see it?
No.
No.
You can't.
Oh,
Can't also see
You didn't
there
change
is
the slide.
Oh, is that so?
Yeah.
Okay.
Okay.
It's
So
okay now.
did you see?
Yes.
So this one, did you see this one?
Yeah, this
Yes,
one.
please.
yeah.
Everything, did you see it?
This one we saw.
Yes,
We saw
please.
this one.
Did you see everything like?
Yeah,
Yes,
this one
Did you
was.
see
Please,
like that?
Yes,
All the
please.
information
let's
I
go.
gave, did you see all of them? Okay,
Yeah,
that's
we
fine.
saw everything.
Okay, that's fine. Okay, good. Okay, so
now you
when
can
I
So let's go to the next one.
okay, good. So
Good.
let's see something here. Okay, so let's say now let me project it. Okay, like that. Okay, good. So now there's the future we've entered. Now the future is a beautiful cycle. What is this cycle called? So this light independent phase is called what? It's called Calvin cycle. Okay, and this Calvin cycle was named after a scientist who is called Calvin. Right. So let's see what is going to happen. Now this Calvin cycle is a very simple process. Okay, what is going to happen is that this particular molecule here, which is called what? Ribulose bisphosphate, which is called, the short form is called Rubisco. Okay, that's a short form. We call it so we call it Rubisco. Okay, that is the term for it. Okay, so with this Rubisco enzyme here, is going to be add this carbon dioxide to this Rubisco ribulose bisphosphate. The enzyme here called Rubisco, which is the ribulose what? Biphosphate carboxylase. Okay, it's going to, this is an enzyme. It's going to add carbon dioxide to ribulose bisphosphate. Are you with me? So just like, remember in the case of Krebs cycle, okay, in the case of Krebs cycle, you can see that there was this enzyme that is a synthase. Okay, that's a citrate synthase. That was the enzyme there. In this case here, the enzyme here, in fact, the most abundant enzyme on the planet Earth is Rubisco. Okay, that's a ribulose bisphosphate carboxylase. This is an enzyme. Okay, because every plant has it. It is the most abundant enzyme in the world, on the planet. Okay, it's the most abundant enzyme on the planet. Okay, so the most abundant enzyme on the planet here, which is this one here, don't forget that, will catalyze the conversion. So this is 5 carbon molecule. And I'm adding carbon dioxide to it, it becomes 6. And this 6 carbon, the moment you add is unstable, it will split into what? Into 3 of what? Remember, bisphosphate means 2 phosphates, right? Remember, bi. Look at that. Bisphosphate means 2 phosphates. Is that not it? Hello? Are you there?
Yeah.
Good. So,
Yes.
so remember this particular molecule, this bisphosphate, is what? It's having 2 phosphate. So the moment you add the carbon dioxide to it, so we call this one carbon fixation. The moment they add the carbon dioxide, we are going to get six carbon molecule, and this 6 carbon molecule will quickly split into 2, and that 2 is the word. That's a glycerate 3 phosphate, like that. So from this, the enzyme here, rubisco, okay, rubisco, which is also known as a ribulose bisphosphate carboxylase, okay, will split. This will catalyze the conversion of what? Of the ribulose bisphosphate with what? With carbon dioxide into a molecule, and then that will split into 2, into 3, into 2 of what? Glycerate. Okay, so you can see that this one glycerate 3 phosphate. Okay, like that. Okay, good. And in this particular reaction, remember this glycerates, okay, 3 phosphate here, all right, guys, 3 phosphates like that. Okay, look at what is going to happen here. You see those things that the husband and the wife generated during the days when the sun was hitting them, all right, which is the light phase of photosynthesis, the light dependent phase of photosynthesis. Now, during that phase, when the light was hitting them, they generated energy, ATP. The man was generating ATP from the photosystem 2. The woman was generating NADPH from the photosystem Y, from the photosystem 1. Now you can see that it has paid off here. So from here, they will use what? They will use this one in process of, they use that energy here to generate the triose phosphate. Okay, that they will use it to generate the what? The triose phosphate. Okay, and this triphosphate, that's a 3 carbon molecule. It is this 3 carbon molecule that will be used to generate the hexose sugar. Okay, and the hexose sugar means 6 carbon. Okay, the hexose means 6 carbon. All right, hexose means 6 carbon, which is the what? The glucose. Okay, so they will use this. Okay, look at that. Okay, so let's see the information here. Good. So now the light independent phase, which is called the Calvin cycle, it's involved what? That's the fixation of carbon. It occurs in the stroma. The stroma, that's the liquid part of the chloroplast. And this one, okay, fixation, incorporation of carbon atom, the carbon dioxide. Okay, that's the carbon dioxide be incorporated. That's the fixing of carbon atom in the form of what? That's the carbon dioxide, okay, into an organic molecule. All right. And then remember that what we did, the hydrogen, that hydrogen ion, that what we carried on has been added here. The ATP will also be released there. Okay, so you see the reducing power here. So that whatever we pick from there, from the hard days when the sun was hitting us, now has been used here to build this 3 carbon molecule here. And look at that, this beautiful enzyme. This enzyme involve what? Okay, so works optimally when, now look at that. You look at what we are saying, that this enzyme here, that's a rubisco, which is going to convert this one into what? Into glycerate 3 phosphate. Okay, glycerate 3 phosphate. Okay, all right, which there will be 2 of them. All right, convert it into what? Triose phosphate. That's three phosphate here. All right. And then when we get this this trials phosphate here, that is where we are going to build our sugar like that. Okay, our sugar, which is the glucose. But you see, in the course of building that, our what? Our ribulose phosphate is generated back. Can you see it? We are getting our ribulose phosphate. Look at what is happening. We added it to it. It will go, and then it will release the what? The ATP. You see, look at that. We break the ATP down again. And when we break the ATP down again, then we'll get our what? Our ribulose bisphosphate back. This particular molecule, you get it back. And I say it's one of the most abundant molecule on the planet. Okay, so we've generated it back. So you can see it's a beautiful cycle. Okay, we started with this molecule. It will go here. We'll add one carbon to it, and then we'll split the molecule into these two of that. And then that ATP that were generated, look at that ATP here. We'll use this portion to generate that. Okay, and then when we use it, that's where we are going to get a 3 carbon molecule here. So it is out of this 3 carbon molecule that we will get our glucose, which is the hexose, 6 carbon sugar, hexose sugar. Okay, good. Are you there? Are you there? So the carbon fixation does not, remember, see that the carbon fixation does not occur, that does not occur in darkness, as it is named. So what it means here is that it does not depend on light. Please, you understand what I'm saying? It is light independent.
Yes, please.
So what it means here is that whether light is there or not, it will occur. Are you there? It doesn't depend on, on what? On light. Okay? So whether light is there or not, the second phase of the life cycle does not require light. So whether the light is present or not, the Calvin cycle will still occur. So you see, most of the time, say the carbon fixation does not occur in what? People think that does not occur only in what? Darkness. No. Okay. So what is that? What it means here is that it does not require light. Okay, it doesn't require light. So whether light is there or not, it will still occur. Okay, all right. It is light independent. So whether light is there or light is not there, the process will occur. All right, good. And you see, very important point is here. One very important point here is that the enzyme here, you can see that the rubisco, which is the word, that's the ribulose word, bisphosphate carboxylase. All right. They say they work when the oxygen concentration, okay, when the oxygen concentration in the environment is low. All right. Okay. So this is very important point because I will talk about it later on. Okay. Good. Now, do you know what? Let me take this one for you here. Let me pick this for you here. Okay, let me pick a Calvin cycle for you here. All right, cycle. All right. Let me pick a simple image for you. All right. Let me pick simple image for you here so that you understand it better. So that you understand it better of what I'm talking about. Okay, so that you understand it better of what I'm talking about. Okay, so let me take a simple image for you. Okay, so let's say something simple. I'm coming. Let's see, very simple one that. Okay, so let's see something here. Okay, I'm going to give you one simple cycle so that you appreciate what I'm talking about. Okay, So let's see this one. Okay, so this. Okay. Okay. Now look on my screen. Look on my screen. All right, and see the process. Okay. Look on my screen here. You see there are three processes going on there. Please, can you see that? There are three processes going on there. Okay, so let's see something. Okay, look at this particular process. So there are three processes going on in Calvin cycle. Okay, the first process here is that there is this particular molecule existing within the leaves. And what is this molecule called? Ribulose what? Ribulose bisphosphate. Okay, it's bisphosphate simply because there are two phosphate at the end of this particular molecule. And then there is this beautiful enzyme. We call it what? We call it ribulose bisphosphate carboxylase, and which in short we call it Rubisco. And this Rubisco is going to do something simple. What is it going to do? It's going to fix carbon. So this is what you call carbon fixation. It's going to fix carbon. Okay, so it's going to add the carbon to this. All right, and when it adds the carbon to this particular molecule, the molecule is unstable, so it will split into what? Into 2,3-phosphoglycerate, like that. So you can look at that. When I add the carbon to it, I'm going to get 6. So it will split into 2 of this. Are you with me? Hello, hello. Are you there?
Hi, we are around.
Yes, Doc.
Beautiful. I can't hear my ladies. Where are they? You know me, I'm sensitive to this voice.
Oh,
Where are my ladies? Are you there? Are you there?
yes,
Let me hear you. Yes. I keep hearing the voice of gentlemen all the time, and it seems they're the only people that. Oh yeah. Oh, that's beautiful. My day is completed. That's beautiful one. Okay, great. The man you go paying you. Okay.
Oh, Doc, no, you know they pain us.
Oh, see you. You are crying. You will soon be shot
We are
by the
cool.
ladies. You will soon be shot by the ladies in true of us right now. So let's see something here. And look at the image here. See, so I'm giving the detailed stuff here. Okay, let's look at the detailed stuff here. All right, so that you appreciate it better. Look at what I've put on the board. So this is the Calvin cycle. So the Calvin cycle is actually in 3 stages. What are they? The first one is the fixation of carbon. The carbon should be there. You see, look at that. So you see that this carbon molecule is short-lived. Are you there? This carbon molecule is short-lived. Look at that. Can you see it? Okay, I just want to let me give you like 2 minutes. Look at what I've done on this particular screen. Okay, I want you to see this is the Calvin cycle. Okay, occurring within the stroma of the word within the stroma of the chloroplast. Okay, look at it carefully. There are 3 things happening here. Look at that. Okay, look at that. I just want you to observe something. Now look at the number of molecules here. You see, look at here. You see, we are using 6 of the ribulose bisphosphate. We are using 6 of them. And look at the carbon dioxide we are using. We are using 6 carbon dioxide. Are you there? Yes, look at it. I just want you to appreciate this one. Just take your time. Just look at it carefully and jot whatever you could. I just want you to look at it before I'll come in again. Let me just give you 2 minutes to appreciate. This is the detailed stuff there. So the Calvin cycle has 3 main processes. 1, carbon fixation. All right, and look at 6 molecules of carbon dioxide is entering there. 6 molecules of carbon dioxide going inside there.
Please, Doc.
Hello, I'm listening.
So why the 6 carbon molecules?
Beautiful. This is the question I'm waiting for you to ask. Okay. Now, so why the 6 carbon dioxide molecule? So you can see that. So you see at the end of the reaction, you need to generate your ribulose bisphosphate back. This molecule, this one is here. We need to get it back, right? We need to get it back.
Okay.
So let's see something. So it is that carbon that entered the cycle, that carbon dioxide molecule that entered the cycle. It is that particular those carbon dioxide molecules. They are the ones going to generate the glucose for us. They are going to build this for us, not the new molecule.
Okay.
Have you seen it? Now look at something. I want you to picture something here. Have you seen that the 6 carbon dioxide here? At the end of the day, look at that. So you look at 6. So each of this one. So 6 molecules of this. Look at that. 6 molecules of this will come. 6 molecules of carbon dioxide. So each of this one will pick one carbon each. Look at this. So this is a detailed thing. Now look at it carefully. Okay. So each of this one carbon each. So we have 6 of this. Okay, 6 of the R. Let me just give you RUBP. Okay, all right, 6 of them. Now we'll match the word carbon. 6 molecules of carbon dioxide should pick it up, and then we'll get this word, this molecule here, which is what? Which is the 6. That's 1,6 what? Bisphosphate, but it is short-lived. It is short-lived. It is intermediate. The moment it forms now, so 1 carbon will be added to each of them. Okay, 1 carbon will be added to each of them. Have you seen? This is 5 carbon molecule here. All right, and then we need 5 of them to come, 6 of them to come, and each of them is going to pick 1 carbon each. Look at that. So they pick 1 carbon each. So you can see that 6 here, 6 here. There are 6 of them here, right? Can you see that? Can you see that? Okay. Yes. Just take your time to appreciate the thing. You don't need to know the intricate details of everything here. But what the most important thing, I want you to see that these 3 phases of what? Of the Calvin cycle. That's the most important thing to me. I want you to appreciate it. So 6 of this one of the most important molecules on the planet. Okay, RUBP. This RUBP, 6 of the molecules are coming. Why? Because if you look at the detailed formula for what? For photosynthesis that we wrote here, if you remember, look at that. You see that we wrote 6 molecules of carbon dioxide. Have you seen it here? Have you seen it?
Yes.
Yeah. So you see that we wrote 6 molecules of carbon dioxide. So all the 6 molecules of carbon dioxide, they are entering here simultaneously. They are going there. So this one is picking all the 6 molecules of carbon dioxide. And then we have an enzyme called what? Rubisco. Okay, Rubisco. You don't need to write a full term. Rubisco is fine. It's globally accepted. So this Rubisco is going to make sure that this carbon is fixed into the what? Into the RUBP. Okay, so you can see that the 5 carbon here has become 6 here. But this 6 carbon here, it is short-lived. It doesn't take long. You understand that? It's just like Moses coming from God, and then his face was shining and began to fade. You understand? So it's just a brief moment, just a fleeting moment. Okay? It's just like Ajiman getting $1 million and blowing the $1 million within no time.
Bad boy.
On the Tema chicks, on the chicks in Tema. You understand that? So that 1 million, so he had the $1 million from here. You understand that? Big jackpot. Po, Ajiman. 6 carbon, 6 what? 6 5 carbon molecule here. Okay, that's the 6 of the RUBP added to this. Pearl adjuvant has gotten, but this is short-lived. Short-lived. This, and then this molecule is highly unstable. Will split quickly to what? Look at that, to 12 of this one. Have you seen it? Okay, so to split quickly to 12 of what? 12 of the, okay, we have 6 of this one, to split into 12 of the what? 3-phosphoglycerate. Okay, or we can say that glycerate 3-phosphate. Okay, like that. Okay, like that. Okay, great. So here, look at that. It was split this way into this 3 molecule here, like that, beautifully, like that, quickly, like that. And then that ATP that was actually generated, there will come into the picture. Okay, remember during the light phase of photosynthesis, that ATP are going to break down 12 ATP down. All right, we'll break down 12 ATP down to generate one intermediate molecule, which is 1,3. Remember, you see, look at that. So you see those boys here. These guys are now, they are what? They are phosphates. They are 3, we have 12 of them. Okay, 3 carbon molecules. Okay, and then we'll break ATP down. Okay, the ATP that we gathered, that ATP we gather in life, we'll break 12 of them down so that we will pin this particular molecule down. You understand that? Okay, break this together. So break, so there are 12 of this. We break 12 of them down here to get 1,3 bis phosphoglycerate, like that. And then we'll break another 12 of what the wife generated. Remember, the ATP were generated by the man from the wife, from the photosystem. And then you break 12 of what the wife also generated from the photosystem 1. That's the NADPH from the photosystem 1. And then we produce this beautiful glycerate. Okay, this is the guy. So aldehyde 3-phosphate. That's the 3G3P. And then from this 3GP, will undergo, okay, inside will be released into the what? Into the cytoplasm. And when this one gets into the cytoplasm, it is getting into the cytoplasm in the form of what? So this is the glyceride what? Phosphate. This is the Strauss molecule. So within the cytoplasm, that is where the magic is going to happen. So when they release this 3 carbon, the G3P molecule, and this G3P molecule gets there, that is where it can even be converted into glucose. It can be converted into other molecules. Okay, all right. And then, but during that process, we need to do what? We need to get our what? Our RUBP back. Okay, but look at what is going to happen. Now look at the process. Look at how many of them are we going to get back. Look at the process. Look at how many of them are we going to get back. How many of them are we going to get back? You see, so out of the 12 molecules here, have you seen that only 2 of the molecules is going to form the what? Look at the 2 of the out of the 12 that we generated. Okay, you see the only 2 of them is going to form the what? The G3P. Okay, the Strauss molecule, right? The Strauss, which is going to be used to build up the what? The glucose. All right, but look at the what? The 10 of that is going back to generate what? To regenerate our what? Our bisphosphate back. So if it is 10, now look at that. Look at it. If it is 10 here, and remember each of them is 3. Have you seen it here? So how many of them can regenerate? So you see, you go there and then you break another ATP. Okay, you break another ATP so that, okay, look at that. So we have this 10 here. Okay, so 10 of these 3 molecules here. All right, so you generate 12 here. 10 will go, okay, 10 of them will go into the what? To regeneration, the what? The RUBP. All right, 10 of them will go there, And then in the course of that, we'll break 6 ATP. Okay, 6 ATP will be broken so that we can add the phosphate back and generate our ribolus 1,5-bisphosphate back here like this. Have you seen it? Okay, so if 10 is going here, let's say 10 contains 3. So how many are you going to get here? So 10. So 10 of them here, each of them containing 3. So how many do you think we are going to generate here? Yes. So 10, and I need 5 carbon, right? And 10 are going. So how many do you think we are going to generate?
2.
Now look at that too. Look at that. 10. Look at that. 10 of 3.
Okay.
That
6.
is it?
6.
10 of 3.
Yes.
Is it
That's
30?
30 divided by
That's 30. 30.
Yeah.
And I
divided
want
by 5. That will be 6.
yes. Have you seen it?
Yeah.
So have you seen that? Have you seen that at the end of the day, you are regenerating what you took? Do you get a point, right? Is it that you took 6? You are bringing the 6 back just the way you took it.
Back. Yeah.
Do you understand that? Okay. So let's take it. Let's take our timing. So let me just give you one. Let me just give. This is the most important part of whatever we are doing today, because the other part we'll be talking about is just I've even given a head start already. So that one just like a 15 minutes or something minute and we are done. This here is the most important thing. Okay. So you see how we are getting our 6 carbon dioxide molecule, because we are going to get the R. The inasa, when you write the RUP is accepted. If we instead of ribolus bisphosphate carboxylase and you write rubisco, it's also accepted. Okay, look at the 3 cycle. So we have one particular enzyme is coming here. Okay, that's the rubisco is coming. What is the rubisco is going to? It's going to fix the carbon dioxide that is coming from the atmosphere. We'll fix it, but we need 6 of them. Okay, and we've taken 6 of the RUBP. Okay, let me call it RUB. Okay, RUB. We need 6 of the RUB. Okay, catalyze the rubisco. Okay, to react with our carbon dioxide to give us what? To give us this one will give us 1, 3, 1. This one will give us what? This one will give us 1, 3 bisphosphate, right? If you remember, okay, 1, 3, okay. This will give us 1, 3 bisphosphate. Okay, this is this carbon molecule here. Don't worry about this one. This is intermediate process, so we don't even talk about it. It's very shortly. The moment it's formed, it will split into 2, but we have 6 of them. So when they split into 2, we are getting our 3 of what? So 3 phosphoglycerate. But you see, this is 6. So when they split into 2, we are getting 12 of them. All right. And then we are going to break the energy that we had at the light phase of photosynthesis. We are going to break that energy, ATP. Okay, because we have the 12 of this molecule. We'll break 12 ATP down and we'll attach it to each of them. So now we are going to get what? So this molecule, we are going to add the phosphate to pin it down into the cycle so that you don't run away. All right? Just like the way just like the way side chicks can pin bad boys like Ajaman down. Okay, so always we break the ATP here to pin this molecule them down. Do you know what? I don't want to go anywhere. I want to pin you down. Okay, and I trust my ladies for that. Okay, they will do fine job. Okay, all right, good. So remember, look at this particular process. You see, this particular process is very important. ATP is used, but the most significant thing occurring there is reduction. Okay, look at that. We are reducing this particular. There's reduction. Okay, and what is reduction? Reduction is basically addition of what? Hydrogen. Okay, all right. And this reduction process here requires ATP. Okay, so you can see that all the ATPs that we generated from the what? From the light dependent phase of photosynthesis within the membrane of the what? Of the thylakoid. Okay, now we are using them here in the what? In the reduction phase of the what? Of the carbon life cycle. So that we are using the what? All those energy here, we are using in the reduction phase, and we are using in the what? In the regeneration of the RB, the ROP. Okay, so at the end of the day, you realize that the 6 that you took, you are going to bring the 6 back, because you see, when you get here after the reduction, when we pin this particular molecule down, we added the phosphate to pin it down, then we reduce it. The most important, one of the most important molecule, that's a aldehyde 3-phosphate, which is the triose molecule. That's the triose carbohydrate we've been talking about. So this triose carbohydrate will be released into the cytoplasm, where this glycoaldehyde 3-phosphate will be used to generate glucose and other molecules. All right. So the Calvin cycle, what it does, it doesn't generate the glucose directly, but what it does is that it builds the precursor of the glucose. You understand that? It's just generates. So we have the 2. So you can see, look at these 2 boys going here. Okay, you can see 2 of the what? 2 of the glycoaldehyde phosphate, and we know that when we join these 2 of the glycoaldehyde phosphate, we are going to get our glucose. Please, can you see that? When we join the 2 of the glycoaldehyde phosphate together, we will get what? We will get 6. Okay, when we join these 2 together, we will get 1, 2, 1, 2. It will be 1, 6, 1, 6. No, it will be glucose 1, 6 bisphosphate. Remember, to be glucose 1, 6 bis. When we join these 2 together, remember during glycolysis, when we break the glucose 1, 6, glucose 1, 6 bisphosphate together. Okay, when we break it together, we get what? We get the glycoaldehyde 3-phosphate. If you could remember. Okay. Do you remember? In glycolysis, when we break the glucose 1, 6 bisphosphate together. No, we break the fructose 1, 6 bisphosphate together. We break that one, okay, together, and we're going to get the what? Our glycoaldehyde 3-phosphate. Because in the glycolysis. Hey, the mothers.
Hey.
Are you there? Hello.
Hello.
Okay.
Yes.
Hi.
So these 2 molecules. So when we join them, we eventually generate glucose 1, 6 bisphosphate, and eventually they will take the phosphate off, and we'll get our glucose. Are you there? So some of the tricky true or false question I can ask you is this: that in the Calvin cycle, glucose is generated directly from the Calvin cycle. True or false?
False.
False.
Excellent.
False.
Okay. The Calvin cycle has 3 main phases. What are they?
Carbon
Carbon
fixation,
Excellent.
reduction of
Reduction of
3, regeneration of Ruby.
3p2.
Excellent.
The
Excellent.
Regeneration of
This is all that you need to know. Beautiful multiple choice question. I read the following in order of the cycle, as shown in the Calvin cycle. Don't forget that carbon fixation comes first, followed by reduction of 3-PGA to G3P. And 3GP is the one of the most important molecules. And 3G, remember, G3P is what we are referring to as the triose molecule. That's a 3-carbon molecule that will be generated. And this 3-carbon molecule will be released into the cytoplasm. And when it gets into the cytoplasm, we can use it to build a lot of molecules that we enumerated here. We use it to build a lot of molecules that we listed here. I think I've chopped off that part. Okay, I've chopped off that part. Okay, we listed. Okay, good. So the cycle is in 3 main phases. What are they? Carbon fixation, catalyzed by an enzyme we call Rubisco. And then we have the what? We have the reduction of what? 3-PGA to G3P. And then the last one, regeneration of ROP. That is all. This is what you need to put in your head. And the G3P is what will be released into the cytoplasm that is going to build up our glucose and other molecules. Are you there? Like that. Okay? Like that. Okay, so it's very, very simple. Very simple. Okay, good. So the 6 ROP that you picked, you pay it back. Okay, you pay it back. It's very simple. Okay, all right. So please, let's see whether our ladies can get this one. Okay, so here I'm just giving you just 2 minutes. Okay, on this one, and then I'll give you some simple true or false question here. See whether you can get it. Okay, all right. Just look on my screen. Yes, hello.
We are waiting.
Oh, I trust my ladies. Look at the way the voice alone. The ladies are saying we are waiting to match the men boot for boot. Oh my God. Ajuman is going to go down. Somebody is going down. Ajuman, get ready. Okay, good. Now, let's see here. Okay. Now, number 1. Yeah. Number 1. Okay, so the number 1 here. Good. Number 1. Having cycle is an hexagonic process. Number 2. Number 2. The excited electrons from photo system 1 is replaced by photolysis of water. 3. The Calvin cycle generates the glucose directly. 4. Rubisco enzyme works well at a high concentration of oxygen. Ten molecules of G3 PGA is used to generate 10 molecules of this are directly used. 10 molecules of 3-PGA are used directly to generate 5-RUBP. 6 Calvin cycle cannot occur in the absence of sunlight. 7 Calvin cycle cannot occur in the absence of sunlight. The first step in the Calvin cycle is the reduction of 3-PGA to 3-P. Hey, our mothers are in Sunday school.
This is Victor Kinky Man. This is Victor Kinky Man.
Victor, where are you?
No, come here.
Oh, so you are causing all the troubles in this world, and I'm mentioning our mothers.
No, I don't know that my mic is on. My friend are warning me this morning. I'm in the school.
Oh, okay. That's good. What a good citizen you are. Oh, so that's nice. Okay. Electron transport chain and chemiosmosis occur in both respiration and photosynthesis. The hydrogen carrier in the light, the hydrogen carrier in the light dependent phase of photosynthesis, the hydrogen carrier in the light dependent phase of photosynthesis, in the light dependent phase of what? In the light dependent phase of photosynthesis is NADH. Ten carbon fixation occurs in the thylakoid lumen of the chloroplast. In light dependent phase of, in the light dependent phase in the light dependent phase of photosynthesis, in the light dependent phase of photosynthesis, in the light dependent phase of photosynthesis, ATP, hydrogen ions, and NADH are produced. Unlike photosynthesis, unlike photosynthesis, unlike respiration, photosynthesis involves photolysis of water. Involve photolysis of water. Unlike photosynthesis, unlike respiration, the Generation of ATP. Generation of ATP requires ATP synthase. In cyanobacteria, photosynthesis occurs in the chloroplast. Carotenoid absorbs usually, okay, let me make it. Carotenoids usually absorb red. Carotenoids usually absorb red, orange, and yellow light of the visible spectrum. Start work. Okay, so my ladies, please make sure you show the guy something. Okay, show the guy something, please. No matter what, show the guy something.
We shall show them Pepe.
Oh, see you. You go cry it's all end in tears. You go cry very soon. When the likes of Samuelina, Belinda, okay, Juliet, the Macy's, okay, when the likes of those people are here, my ladies, where are you? Are you there?
Okay, we are there.
I know. Gosmon, the likes of Gosmon.
Sir, please, the 15, is it? Sir.
Carotenoids usually absorb. Oh, so I can't think absorb. Oh, when I make mistakes, only the lady will see it. Oh, what? Oh. We thank God for creating ladies. So Sophie? Sophie?
Yes, Doc.
Okay. My ladies, are you there?
Yeah, there. We almost done, sir.
Good. I like the way you are matching the men boot for boot. Life is better that way. Ajuman, are you done? Or you are still you are still in number 6?
Number 15.
Okay. I'm waiting for my ladies to finish.
In fact, today my network is worrying me very, very well.
Oh.
Network is disturbed. Very.
Tema
It has not
of
been
all
stable.
places. Tema of
Yes,
all
so
places,
network
you have better network.
it's not stable at all, especially if you're not using MTN. It's kind of rough.
Oh,
But
okay.
we are man.
Okay. All right. So let me take this top one away and add my last 5. Oh. Okay. So because the numbers are automatic, that is why. But don't worry. Okay. So all right. Let me see. Let me go back. Okay. So let me take the first 10 away. Okay. Here. So what I'm gonna add is 16. 16. 16 here. Photosynthesis is water is released during photosynthesis. Seventeen. The liquid a group of okay, so a group of thylakoids forms a group of grana forms the thylakoid. At the end of Calvin cycle, at the end of the Calvin cycle, 3 PGA is converted into glucose.
Yeah, okay.
Number 19. Number 19. The correct order of the Calvin if a plant leaf takes in 18 molecules of CO2. How many 18 molecules of CO2 molecules of CO2, 9 molecules of G3P will be released
Mr. Ajiman,
into
mute your
the
microphone.
cytoplasm.
Oh,
And then 20.
sorry.
20. If a plant leaf takes in 24 molecules of CO2, 24 molecules of CO2 for carving. Listen, let me say for carving cycle. For carving cycle. 2 18 molecules of RUBP will be regenerated. All right. Okay. You getting ready to stop work?
24. Number 20.
Yes.
I don't understand the sentence. They say if a plant leaf.
If a plant leaf takes in.
The takes in is the okay.
Yes, takes in 24 molecules of carbon dioxide for carving cycle. 18 molecules of RUB will be generated. That's the ribulose 1,5-bisphosphate will be generated. All right. Safini?
Amen. First to pick more force. I be no doctor that.
Oh, see you.
Most of the answers will be false. So if you pick more, yeah.
All right, can we start?
Obiba Besun.
Oh, people are
Yeah,
coming
doc,
to
you can
cry.
start.
Please.
Okay, we can start. My ladies, are you there? My ladies, are you there?
This time around, they are going to fail. They are there. Men too, we are here.
Where are they? My ladies, are you
Obiniyem. Obiniyem.
there? Oh, my ladies, are you there? Charlotte and Co. Anita, are you people there?
We are there.
Okay, beautiful. Belinda, good. All right, good. Let's see. So we are going to mark.
Dog, we are there.
Great.
We there.
So number one, Calvin cycle is an hexagonic process. True or false?
False.
Okay, so hexagonic means that it gives energy. All right. Okay, so it doesn't. Hexagonic means that it will release energy in the form of ATP. Okay, so that is false. Good. Excited electrons from photosystem 1 is replaced by photolysis of water. That is what? False. Calvin cycle generates the glucose directly. That is also false. Rubisco enzyme works well at a high concentration of oxygen. That is false. 10 molecules of 3-PG are used directly to generate 5-PDES. That is false.
False.
Because it is this one. It is they use 10 molecules of G3 directly, not this one. This one will be reduced to what? Okay, this one will be reduced to the glyceraldehyde 3-phosphate. Okay, so it is this one that is used. It's the G3P that is used directly, not this. Okay, all right, great. Calvin cycle cannot occur in the absence of sunlight. That is false. And then the first step, oh, I didn't mean the step here. The first step in the Calvin cycle is the reduction of 3-PG to G2 there. That is also false. Electron transport chain and chemiosmosis occur in both what? Expiration and photosynthesis. That is a true statement. Hydrogen carrier in the hydrogen carrier in the light dependent phase of photosynthesis is NADH. That is false. It is what? It is NADPH. Oh God. And then carbon fixation occurs in the thylakoid lumen of the chloroplast. That is a false statement. It occurs in the stroma. In the light dependent phase of photosynthesis, ATP, hydrogen ions, and NADH are produced. That is false statement. It's NADPH. Unlike respiration, photosynthesis involves photolysis of water. That is a true statement. Unlike respiration, the generation of ATP requires ATP synthase. That is false. In both cases, this one, ATP synthase is required in both cases. So that is false statement. In cyanobacteria, photosynthesis occurs in the chloroplast.
False.
That's
False.
false. Carotenoids usually absorb red, orange, and yellow light from the visible spectrum.
That's false.
False.
That is a false statement. That is
Right, it's rather.
false statement. Okay, good. That is a false statement. Okay, let me reduce this one here. That is a false statement. So this one is a false statement. And then number 16, photosynthesis. Water is released during photosynthesis. True or false?
False. False.
It's true. This is a very tricky question.
False.
You've forgotten when I show you this one, this particular image. Water is released. Okay. Oh, sorry about that. I hope no lady had it wrong.
Yes.
No lady should have no way.
Wrong.
Oh, the ladies who had that one wrong, I'm sorry. It's all my fault. Please forgive me. I'm on my knees begging. Ladies don't deserve to miss that question. I'm sorry, please. You don't deserve that. Please, I'm sorry for that. A group of grana forms the thylakoid. True or false?
False.
False.
So in the word, at the end, I didn't bring the end. At the end of the Calvin cycle, 3PG is converted into glucose.
False.
True or false?
False.
That's false. It's the word. It's G3P.
G.
3P. It's the G3P that will be converted into G3P. That's the glyceraldehyde phosphate. Okay. If a plant leaf takes in 18 molecules of carbon dioxide for carbon cycle, 9 molecules of what? 9 molecules of G3P will be released into the cytoplasm. True or false?
True.
No.
It's false.
If I take 18, 6. When you pick in 6, 2 will be released into the cytoplasm. If you take 6, 3 will be released into the cytoplasm. No, if I take 6, 2 will be released into the cytoplasm.
Yes.
Are you there? When I pick 6 of the word 6 carbon dioxide, okay, if I pick 6 carbon dioxide, I will end up producing 12 what? G3P. And then the 12, 10 of them will go for regeneration of the RUBP.
UBP.
And then 2 go into the cytoplasm. So if I pick 18, everything will be times 3. So it is going to be 6. So here is a false statement. One man is down, one man down, one man down, one man down. So it will be 6 words. 6 G3P that will be released into the cytoplasm. Question number 20. If a plant leaf takes in 24 molecules of carbon dioxide for carbon cycle, 18 molecules of RUBP will be regenerated. True or false?
False.
False.
That is false. How many will be regenerated?
It
It will be where?
12.
It will be the number of carbon dioxide will be equal to the number of that will enter into the reaction. So if 24 molecules of carbon dioxide enter the Krebs cycle, it means that we are talking about 24 molecules of RUB that will be used in the Krebs cycle. So here, if it is 24 will be generated. The number of carbon dioxide will be equal to the number of RUB that will be used. So at the end of the day, you regenerate all of them. So it will be 24. All right. So who had 20 over 20? Who had 20 over 20? Yes, we are 20/20. Okay, so Anita 17/20. Oh, my lady, so proud of you. I know my ladies will make me so proud. Okay, Anita 17, that's splendid. Ajuman, your 18, your 18 will be subjected. Oh, Valencia, 18/20. That's beautiful. My ladies are on fire. Bashir, what did you get? 19? No, Ajuman, Bashir had 19 though. This one. Yana, this one we
Why?
need
Why
to.
are you
Yeah, I
doubting that?
have
Anna, you're
Hold on.
19/20. Oh, Prosper, well done. 18/20. That's good. Oh, my Juliet. Oh, my Juliet. Oh, that's good. 18/20. Oh, Belinda, I see you. 16/20. That's splendid. Bashir, your 19/20 will be subjected to pink sheet scrutiny. Pink sheet scrutiny at night.
Hold on.
It's not true.
Bashir, this one, High
Hold
Court
on.
will come. No, I'm not doubting you, but we have to subject you to pink sheet scrutiny.
Okay, no problem, Doc.
Okay,
I'm
great.
ready to defend that.
Oh, that's beautiful. That's beautiful. I trust you for that. Okay. Oh, Fidelia, 14 is not bad. Oh,
We
Anna,
are ever ready for forensic audit.
oh, I see you. I see you. Anna,
We are
17,
ever
you
ready
are
for
there.
forensic audit
Nailed
of our
it.
marks.
Oh, Irene, give me a wave. Give me a wave. Oh, my ladies are on fire. Let me call the fire service. Oh, the place is burning. The men, what are you getting? You see, the men are scared of writing. They're not writing their scores. Ajimar, your people are not writing their scores. They are scared. Eh?
You are not.
You
are not
always
mentioning our.
subject
That's
our
what
But
marks
I have
I'm
19 over
looking
there.
at it.
I
I'm
swear.
looking at it again. Where are they?
You
Where
are
are
jumping
the men?
over ours, so you have stopped.
Jumping over? I'm here. We're looking at it. I
But
can see
why
Mark
do you always doubt?
I sent mine long time.
Say,
I was the second person, but
say,
didn't
why do you
mention
always
it.
doubt the
I've
men?
seen Prosper 18. Well done. Even though we'll be subjected to pin sheet scrutiny, Ajiman, still yours too. Bashir, pin sheet go come inside, no matter what. Oh, my Anita, 17/20. I trust you. Valencia, 18. This is beautiful. Juliet, 18. Oh, my Belinda, 16. Oh, Fidelia, well done. I realize your next keep one had not been that would have gotten 20/20. Oh, Anna, my one Anna. Oh, 17. Irene, 17. I see you. My Mercy, 15. Well done. Now the gentlemen have started coming. What are they getting? 16. Okay, not bad. Sammy, not bad. Ajuman.
This is my worry.
Victor, 19. Eh?
Hello, Doc.
Victor 19.
Victor.
Hello, Doc.
Yeah, Prosper.
Hello, sir.
Prosper, yeah, I'm listening to you.
Please, I want to put up this argument and see. It means
When
that.
did you finish eating the cake?
Who is saying that? Who is
Sir,
mentioning
it means that. How do we call it? The Calvin cycle. So if the product we get from the how do we call it? The light dependent cycle, they are going into the Kelvin cycle.
The products you
From
get. Yes. You are right.
So,
you are right. So the products you have from what? From the
light.
which cycle? Come again.
The products from the
yes.
light dependent cycle, they are going to the Kelvin. What do we call it? Is it the Kelvin cycle?
Yes, that's correct. That's correct.
So I'm looking at the question 6, which means if the light dependent phase does not occur, we cannot have the other cycle. Am I right?
Yes, please.
So I don't know.
So I get what I come from. So Yes, so the one a particular question. I think you are building a point. One particular question. So if there's no light, let's see. Let's see which question is that? Let's see which question is that.
Is it question 6 or
question 6? Carbon cycle cannot occur. Carbon cycle cannot occur in the absence of sunlight.
So if the first cycle does not occur, can we have the second one?
No, but because I started with a carbon cycle, without sunlight, still the carbon cycle was still okay.
Yeah.
So because it doesn't depend on it.
Okay.
So we don't have to, because of the statement I made. The statement I made is that the carbon cycle, so you don't have to go back and say that, oh, because this one is there and it will not okay. No. Because I started with the cycle. This cycle is light independent. Because I started with the cycle and it's light independent, which means the absence of sunlight will not affect that particular cycle in any way.
So you are talking about that particular cycle alone?
Yes, please. Yes, please.
Okay. Thank you, sir.
Yes. All right.
Thank you, sir.
You're most welcome. Okay, so let me see score from my Joe baby, Josephine, I see you. 18. Pray you are there. I'm even praising God. Hey, Victor, this is your 16. Oh, Ajiman, Alex is 16 number. Victor, this is your.
My score is correct. I had only the 16 wrong.
Yeah, Victor, it will be subjected to still pink sheet subjection will come in. That's what I'm saying. Pink sheet will come in. No matter what, pink sheet will come in. Okay?
Hello, Doc.
Pink sheet. Yeah, hello, Kingsley.
Yes,
let me
please,
hear you. Yes.
Question number 16.
Question number 8. Question number 8.
Right.
18 or 18 rather. Item number 18.
16.
Water is released in photosynthesis. Yes.
Please.
It's released as one of the byproduct. It's released as one of the byproduct. Yeah, it is.
I didn't get this summary very well, sir.
Okay. So this side, this one here, so you can see here in the stroma, it's released. Okay, but you see, because this one is happening in the stroma, in the stroma. The stroma is actually a solution. So that water will be dissolved in the stroma. That's why we don't talk about it. Have you said it? I look at what I have on my screen. You can see that water is about that. The stroma itself is liquid. So the water will be inside the stroma. It's not going to be released out like that. To be inside the stroma. All right. Okay, are you there?
Yes, no.
Yes. Okay. Good. All right. Okay, great. Okay, so we are done with this side. Okay, we are done with this side. Please, do you know what? The last part, I'm going to use 5 minutes to talk about it. The last part, I'll use 5 minutes to talk about it. Okay, please, just 5 minutes to talk about it. Okay, because I've already talked about much about that one already. Okay, now we have something we call photorespiration. Okay, photorespiration is this. Okay, now look at what happens. Okay, look at what happens here. When carbon dioxide, see the one, all those things that we described. Okay.
Hello, sir.
Hello. Yes.
Hello, sir.
Hello. I'm listening.
Please, Prosper and Juliet, if they can mute their mic.
I'm sorry.
Prosper, please mute. Jew, are you there? My one and only Jew, are you there? Please mute your mic for me, okay? All right, great. All right. So here, I'm going to use just one slide and I will explain everything here in 5 minutes and we are done. Okay. So, so when it comes to the, so there's something we call photorespiration. Okay? Photorespiration. All right. Now, photorespiration is very simple. You see, when we're talking, we said something nice. We said something like this, that carbon dioxide, this enzyme here, this enzyme here, this enzyme called Rubisco. Rubisco works very well when what? Oxygen concentration is what? It's low in the what? In the intercellular space. So the oxygen should be low. The oxygen concentration should be low. Do you know why? So oxygen concentration should be low for this one to work. All right, good. Now, so this is how Rubisco works. Now, when oxygen concentration is high, when O2 concentration is high, do you know something? This Rubisco, just like the way men are argument, a married man will still chase the side chicks. Do you know something? Why? Because the side chicks, okay, they have what? What the side chick will do. Sometimes, you know, the sweetheart at home will not do it. You know what I'm saying? Okay. So this one, what happens is that when the oxygen is there, okay, now we expect the Rubisco to pick up the carbon dioxide for the what? For the fixation, to pick up the carbon dioxide. But when there is high concentration of oxygen, the Rubisco has high affinity, love oxygen more than the what? More than, okay, when the oxygen is there, the Rubisco has high affinity for oxygen more than carbon dioxide. Are you there? Are you there? Do you get what I'm saying here? Now, Rubisco is the enzyme.
Come again.
Okay, listen, Rubisco is the enzyme. So let's take it this way. Let me take Ajuman, okay? Because Ajuman is the only bad man here. Now, Ajuman is here. Ajuman is married, okay, with a beautiful wife, okay, and stuff like happy home. Now the oxygen here, now this marriage can do very well only when there is no distraction from side chicks, okay? So this marriage should go very well when the Rubisco only pairs with what? Pairs with what? With the carbon dioxide. Beautifully. Okay? All right. Pairs with the carbon dioxide beautifully. Pair with the carbon dioxide beautifully. But Rubisco tend to have what? High affinity. So Rubisco is the man and carbon dioxide is the wife. Now here we are. This is Ajuman. Now, oxygen is a side chick. You understand that? Oxygen is a side chick. Are you with me? So when oxygen is there, Ajuman will snub the wife and go with a side chick because Rubisco has higher affinity for oxygen than carbon dioxide. So when oxygen is there, Rubisco will rather be oxidized into what? Will rather be oxidized into three carbon what? 3 carbon and 2 carbon molecule. Are you with me? Okay, so when instead of carbon picking up carbon dioxide and then going to what? Going to form 6 carbon molecule that will break down into what? Into that 3 carbon molecule. And so rather this oxygen, in presence of oxygen, this one RBP, this will rather react with what? Oxygen here. O2, like that. Okay, so and then this one will rather what? So it's more or less oxidation will occur to produce what? To end up producing carbon dioxide instead. It's a rather what? in presence of oxygen, this is rather going to occur. Okay, the photorespiration is going to occur instead. So this, whereby this, in the presence of oxygen, this rub, okay, this one here, they will compete for the active site. Have you seen it? So here, this one, the active site of this enzyme has high affinity for oxygen. So this is the enzyme here. So in presence of that, they rather combine with oxygen to release what? Carbon dioxide gas to be released. And 3, okay, so instead of this carbon going to form what? Going to form the 3 carbon molecule, 2, 3 carbon molecule. It will end up going to form what? 1, 3 carbon molecule, and 2 this. And this one would deplete, it will reduce the what? The efficiency of what? It reduces the efficiency of what? Of photosynthesis. Because instead of making use of the carbon dioxide, okay, look at what the plant is doing. It's now using oxygen, because there's high oxygen there. It's now changing the oxygen rather, and then changing it to what? To carbon dioxide. And this is what we call photorespiration. And it occurs when there's high concentration of what? Carbon dioxide. And you know, it's also okay when temperature is high. You know, at high temperature, what happens? At high temperature, the stomata will close. The stomata will do what? The stomata will close. And when stomata closes, okay, what will happen is that the carbon dioxide in there will become low. Carbon dioxide in there will become low. And when carbon dioxide in there will become low, what will happen? Oxygen will go up in there. And when oxygen goes up in there, that is where the photorespiration occurs. And so you see, photorespiration occurs when temperature is high, when there's high temperature, and then the stomata closes. And that is what the photorespiration occurs. All right. So this photorespiration is what? It reduces the efficiency of what? Of photo. It reduces the efficiency of what? Of photosynthesis. All right. It reduces the efficiency of photosynthesis. So instead of what should go into the formation of carbon, generating the 3 carbon, this one is now being converted this way. Okay, so photorespiration, and it occurs in what? It's very common in what? Plants we call 3C plants. Okay, and that C3 plant. Remember C3 plant because of the C3 plant are the normal plants we see around. C4 plant. C4 plant example is what? Is the example? We have the word. Can you give me an example of C4 plant? We have the maize,
The legumes.
the legume, and then the word.
The monocots.
Okay, the millets, and then the word. This thing. So this one, like this one. Okay, this one, like they have mechanism. You can see that this one, they do very well in northern Ghana. Do you know what? Look at what this mechanism is using. So this one is now using different mechanism altogether. Okay, it's using different mechanism altogether. So the C4 plant, inside it, you see inside the C4 plant, you see, look at that. You see the C4 plant, you see it separates the what? It separates the carbon dioxide, the rubisco. Okay, the oxygen doesn't get into contact with this one. So the C4 plant, that's where the what? That is where. Okay. Hello, there. Okay, good. I'll be there. So C4 plant has gotten mechanism of what? Of making sure that this rubisco, which will love the what? This thing more, will rather will not go through that process. So look at the mechanism it's going to use. So look at the C4 plant. So you see, so this those C4 plants. So in high temperature areas like northern Ghana, you can see that the C4 plant, they can do very well there. So northern Ghana, when you plant maize and millet and sorghum in northern Ghana, high temperature areas, they can do very well. Why? Because they have mechanism of separating this one. Okay, they have mechanism of what? They have mechanism of what? Of preventing this one. They will generate. They have the ability to generate the what? Unlike this one, that the C3 plant generates what? That's a glycol 3 phosphate. That's the C3 plant. Okay, but the C4 plant will generate the oxaloacetate first. And this oxaloacetate first is able to bypass the what? So it doesn't use it uses different mechanism, it uses different enzyme. Enzyme we call it what? We call that enzyme we call pep what? Carboxylase. Okay, it uses the pep carboxylase. Okay, carboxylase. All right, so it doesn't use the word. So the C4 plant like maize and then sugarcane and the sorghum, they bypasses the what? This one, the rubisco. Or rubisco loves the word oxygen more than the carbon dioxide. But in the C4 plant, they will generate C4, which is the what they will generate this particular molecule first. And this particular molecule will what? Will use this enzyme. So it will bypass the what? The rubisco, okay, and generate this carbon 4 molecule. Okay, the oxaloacetate first. Okay, before. And so because of that, it's able to thrive very well in a place where temperature is what? In a place where temperature is high. Okay, in a place where temperature is what? High. And those places, you can see that when you plant in northern Ghana, if you plant those ordinary plants here, the one we'll be planting in southern Ghana, they can't do very well there. But in northern Ghana, if you plant maize, millet, sorghum, sugarcane in northern Ghana, they will do very, very well in those places with very high temperature. All right, okay. But when you plant it, so high temperatures, C4 plant has a way of going around it because it will not use the rubisco. Okay, it will not use the rubisco. It will use the what? It will use the pep carboxylase to do it. Okay, all right, good. So that is how come the what is able to do so to bypass. Okay, to bypass the mechanism first. Okay, all right, and then inside here is where it's going to work. Okay, so the rubisco would not. So the carbon dioxide will not be direct here. So they increase photosynthesis by separating the rubisco from the what? From the oxygen. So it is somebody who will come and so this one here, this oxalo will now bring the carbon here. Okay, so you pick this one, we'll pick it, and later we'll bring the what? So the carbon dioxide, the oxygen doesn't come into contact. All right, so it doesn't use this one directly. Okay, they've separated the two. Okay, so inside there is what is occurring. So you can see this, look at the outside here. You have the bundle sheet outside here. So you can see that they will first of, instead of generating C3 for no, they will generate what? They will generate this thing, C4 first, and then the C4 will come and give it carbon to rubisco. Okay, so they don't use this one first. Okay, no, they will form C4 first, and then C4 will come and release the carbon to the rubisco. Do you understand that? So that it doesn't occur directly. So this one. So the rubisco is not in direct contact with the oxygen. Okay, all right. So that is why they are able to overcome this concept of what? This issue of. So you can see here that surrounding here, this side is where the what? The photosynthesis stuff, the carbon cycle is going to occur, but they are not in direct contact with oxygen. The oxygen will be here, will be in this space. Okay, so this side here is what is going to pick up to form the oxaloacetate, and then that 4 carbon molecule will now come and release the carbon to this one rather. I'll come and give the carbon to you, so you pick it there and come and give it to me. Okay. So that's what happens. So please, I have another class right now. Okay. Go through this one. Okay. And then before you write your exam, I'll try and organize one tutorial class for you. Okay. So that you'll be able to go through. So you can see. Look at the carbon cycle is occurring here. But look at that. You see the first molecule that formed was the azonoacetate will form first. Okay. And then it will release, okay, through that process. Okay. All right. Okay. You can see that. You see, look at that. Kept carboxylase. Okay. Then before it will be released to the carbon cycle here. All right. Okay, great. So please read this slide. Let me save it quickly here and then email it to you. And then, as I said, I will try Ajman. Let me email it to you right now, okay, before it's too late. Okay. Compose. So Ajman. Okay, so Ajman, please, I'm sending the slide to you right now. Okay, so I'm sending the slide to you now. Okay, so let's say, all right. Okay, so I'm coming. File, save as. Okay, I'm coming. Just a minute. File, save as on desktop. Okay, so that's this part 3. Part 3. Okay, so save. Okay, so please go through it, and then the last part I will try, and then in the tutorial class before you write your quiz, please I'll try and then wrap up that one with you. Okay.
Please, Doc.
Yes, please.
You're most grateful, but can we get some
Thank you.
areas for the first quiz?
Oh,
Or...
please read everything that I've taught
Okay,
you.
Doc.
Please read everything. I've given you trial questions and everything. So please read everything that I've taught you. Okay. I've given you true or false questions. So all the true or false questions will be on the slides. Just go through them. There won't be nothing. I don't think it's going to be difficult, different from what you are having. Trust me. Okay, just go through the slide, go through the trial questions we've been doing, and I think you'll be fine. Okay. So Ajiman, please, I'm sending it to you now. Go through it and then okay. Ajiman, are you there?
I'm here, sir.
Okay, so it's loading. So I've hit send, so it's loading, and then the slide will be sent to you. Now, and then you will do this thing.
All right.
Okay, you forward it to your class. Okay. So on this note, I would like to say thank you to you for your time, and then enjoy the rest of the day, and may God richly bless you. Okay.
Thank you,
I'll check
and
them
God
mail them
bless
down.
you
Thank you
No
too.
so
need to.
much, Doc. May God bless you.
F. Robert.
Prosper.
Hello.
That's your phone number.
Follow. How are you,
More time for.
you? Akwenamo.
If I'm not to go Sunday, man.
Ke Sunday mo.
But you had the phone number. It told me, "Boy, me away happy."
Ne de mi west cho. O enyo mi aton ya solu mi fa gake.
They shot you.
How for do? Yo. Okay.
I'm a guy.
Yo.
The camera,
Yo, nenyo no, huh? Bye bye.